All righty, away we go. So this will be our first lecture. Today is March 19th. Welcome everybody. As you see, our goal now, after the fun and harrowing experience of our first online exams, is going to be to move on to our fourth section of the class. We still have not been given a return date yet. They're still leaving it open-ended, but this section is a relatively long section which will take us through spring break. So hopefully we'll come back to the classroom after spring break and we won't have to do another online exam. There was a notice I sent out via email today that Jeff Chingaris is still going to be doing a open lab on Fridays. It's obviously a virtual on lab. You can't go on campus. But he does have some models to show and he will be able to answer questions. So if you want to take advantage of that, you have that opportunity as well. As you can see from our schedule, and I've had several questions on this, so I want to make sure that we cover this. You are still going to be completing and turning in your unit reviews. This section has two of them, units 10 and units 11. However, let me grab that real fast. For those, I haven't given you a set due date for them yet, because what I don't want to do is be inundated with emails. I'm hoping that once we're given a timetable of when we'll return, or if we will return for that matter, then we'll be able to turn them in person like we normally do. If not, then what I will do is I will set up a Dropbox on Canvas so that you can take pictures of them or convert them into PDFs or however you want to do it that way and turn that in. It is still, as it always has been, it's basically an excuse to study this material. So I do want to strongly encourage you to be able to do that so that that will help you to understand the material. The other activity that you're going to have is that PhysioX. I know we had a tremendous amount of fun with it last time, so you will be doing that again. And it is going to be due on the 14th of April, so we have a little bit of time. It's near the end because there is a lot of physiology on this one. What I will tell you about our muscular system is we have just as much anatomy as we did with the previous one, but there's a lot more physiology. The physiology in the skeletal system is very straightforward. This process, the process of how a muscle contracts, is our longest physiological process. Doesn't mean it's the hardest. It isn't necessarily the hardest. It just has a ton of steps. It has so many steps, in fact, that we are going to divide it up into 3 different segments. We're going to do 3 different parts. And whenever we have our next exam, whether in person or online again, I guarantee that 2 of those 3 steps will be essay questions on the exam. So it is a big, elaborate process. Not necessarily hard, but it is elaborate. So be prepared for that. And then one of the things that will help you to understand that is the PhysioX. Back in ancient times, when we had class on campus, and for that matter, when I was a grad student, when we were studying the muscular system, what you would do is you would go and get a frog. Right? They'd bring a big bucket full of them into the classroom and release them into the classroom, and you had to catch one. Once you caught one, you then had to pith that frog, which meant you had to take a large gauge hypodermic needle and insert it at the base of its skull and run it down the spinal cord. Once you ran it down the spinal column, that destroyed the spinal cord. It was paralyzed, but still alive. You could then do things like open it up to study the cardiac muscle of the heart, or you could peel the leg of it, the skin off the leg, and inject it with electrodes and stimulate it that way so that you could see how the muscles work. And as tremendously fun as that sounds, we get to do it virtually. You don't actually have to virtually catch and pit the frogs, but we do have some muscle that you're going to get to play with. There are 7 activities for this PhysioX exercise, so it does take a bit of time. So while I am leaving it to you till the end, you are going to need to spend some time on it. You're going to have to turn it in online in the Dropbox the same way you normally would. But you also don't want to necessarily start on it right away. You want to have some time for us to talk about the physiology so that we'll have the opportunity to do that. All right, real quickly, I think we have a lot of people here before we get started into lecture. You have basically 2 ways that you can interact with me. 3, actually, I guess. The first is you are welcome and encouraged to use your microphones to talk or to ask questions. All I ask is that if you have your microphone hot, that again, be careful of the ambient noise that you have around you. If you don't want to speak, then one way you can get your attention using the participation list, you can do things like raising your hand or giving me some kind of notification or something like that, that you have a question. There's a raised hand. So can I get everybody to go ahead and raise a hand for me using that so that I can see that you understand how to do that? There you go, we're getting there. About a third of you have it, about half, excellent. Perfect, a couple more need to figure that out. All right, and then the third way is there is a chat window. There is a group chat that you can use as well. So you can type questions there or type answers to questions there as well. Raising your hand is a good way to try to get my attention. If you don't want to speak up, obviously you're welcome to speak up as well if you have that. And then, or you can just type a question onto the Zoom chat as we go through it that way. We are gonna go through the slides. And when we go through the slides, we also have a whiteboard here, actually my iPad, where I'll be able to do some of the drawings so that will help us to understand these processes as well. Our goal for today is going to be to talk first about the characteristics of all muscle tissues. That's going to be what we primarily focus on. And then from there, what I'm going to do is I'm going to mute everybody right now. But again, if you want to use your microphone to ask questions, you can unmute it to be able to do that. I'm getting a fair amount of feedback right now. We're going to talk about the characteristics of all muscle tissues. Once we understand the characteristics of all muscle tissues, because we already talked about epithelium, we already talked about connective tissue, and way back in the day, we said when we got to the muscular system, we would talk about muscular tissue. So we're going to talk about that in all of the muscular tissues. After we do that, we're going to spend pretty much the remainder of our time in this section talking about skeletal muscle tissue. And learning everything you ever wanted to know and more about that. And then pretty much on the very last lecture, we'll go back and talk about, yeah, hold on one second. I'll answer that question, great question. We'll go back and talk about smooth muscle and cardiac muscle and how it's different. So yes, I have started the recording when I first started, so you do have the opportunity to go through this, and I will be posting this lecture on YouTube. I was able to do that with the test one we did earlier this week and yesterday's for my 431 class. So again, you can go to our YouTube page to be able to do this. I encourage you to take notes, look around and watch the things that we talk about on this. But yes, you can go back and watch it and listen to it again. All right, any questions on that before we get started? All right, so that is going to be our game plan. So let's go ahead and switch gears now. And come on, there we go. Like I said, our goal now is to start our discussion of the muscular system, and starting first by discussing muscle tissue. Oh, I need my, there we go. Sorry, just getting my stuff in order. All right, so let's talk about the functions of muscle tissue. When we think about the functions of muscle tissue, and this is something we have talked about before, switch back to my spotlight. Obviously, our goal is to use muscle tissue to produce movement in our body, move our body through space, right? We've always talked about the importance of movement. We just finished talking about that with the skeletal system, how those bones and those joints act as the levers that our muscles pull on to be able to move us through space. But, and this is normally where I would stand in the middle of the classroom, I would close my eyes, I'd put my arms out, I'd hold my breath, and I'd stay as still as I possibly can. And I would ask you, do I have muscles contracting? At which point you would, of course, say yes. One example of that is as you're trying to stand there and hold your posture, your body is making a whole bunch of tiny little minute contractions that help to stabilize and hold your body in place, right? We take this for granted until that is impaired, right? If now that we're all stuck at home, you like me have become an avid day drinker, right? Then after that 4th or 5th Long Island iced tea, you have a little problem standing upright and coordinating those tiny minute little contractions of the body that maintain your posture. Another example is, again, as you're hopefully sitting during this lecture, your leg or your knee, for instance, isn't moving too much. The non-dominant arm, your shoulder, isn't moving too much. But when you're not using your left arm, the humerus doesn't droop out of the socket of the scapula. We have constant contractions of those muscles that are stabilizing the body positions and holding our joints in place. Of course, the other movement that's going on while I'm standing there with my arms out, my eyes closed, and holding my breath is that we are changing and moving substances in our body. Our heart is beating to pump blood to all parts of our body. Our digestive system is working on that cheeseburger you had for breakfast and moving it through our body. So we are having all of this movement that is going on internally inside of our body. And as we've talked about, what is it, of course, that makes the world go round? This is the part where you respond, either verbally or by typing.
Pressure.
There you go. Absolutely. It is pressure that makes the world go round. And so what we do, whether it's the heart or whether it's the stomach or whether it's the small intestine, we contract those muscles, that skeletal, that cardiac muscle or that smooth muscle, and that changes the volume of our organ. And when we change the volume, we change the pressure. And when we change that pressure change moves substances, whether it's the blood, whether it's breakfast, through our body. So again, we have all this stabilization, we have all this supporting of our joints, holding things in place, moving things internally, right? And not just the movement of our body in space. But there are a couple other important functions of our muscular system as well. One of the things we talked about way back in the integumentary system, when we were talking about the skin, is we talked about how shivering, or heck, we even talked about it on the first day when we talked about homeostasis. Shivering is an important way that our muscles help us to maintain our body temperature, right? There's that key word, and we'll talk about it a lot in this class, but I'll write it here as a reminder. Oops, this helps if I spell it correctly. Oh, goodness. And that is a motor. Remember, a motor is something that we talked about basically converts chemical energy into mechanical energy. In the case of the cars, your car engine does that. Our muscles do the same thing as well. In the case of our muscles, the chemical energy is, of course, ATP, and then it's used to make all these contractions and these movements that we talk about. Like the engine of your car, our muscles, those motors, are not 100% efficient. In fact, they lose a fair amount of the energy in the form of heat. I think we talked about way back when we talked about homeostasis. When certain muscles of your body contract, as much as 75% of the energy, in fact, is lost in the form of heat, right? That wouldn't make a very efficient car engine. But the one thing that it does help us with as an advantage is that that heat is used to maintain our body temperature. So it is definitely beneficial for us that way. Lastly, right, now that everybody is hoarding toilet paper but not doing so good on the food standpoint, right, we're all going to be on the coronavirus diet where we get to have a piece of popcorn for breakfast, a piece of popcorn for lunch, and then 3 pieces of popcorn for dinner, right? And if we were to do that, because we're hoarding all of our foods and everything else for how long we're going to be here, we will definitely lose weight. So when we come back 4 weeks from now or 6 weeks from now or whenever it is, we're all going to be nice and thin and be all ripped with muscles, and all the fat's going to be gone, because that's what gets lost. And when you starve yourself like that, no, of course not. When you starve yourself, what your body actually does is hold on to the fat. Adipose is a treasure. Adipose is prized. So when you starve yourself instead, what your body actually does is it breaks down muscle. Whoops, I got to switch back to... Let's start with the mouse for now. It breaks down muscle. Muscle, as we're going to see, because we're going to look at the microscopic anatomy, is filled just to the brim with proteins. Chock, chock, chock filled with proteins. And so when your body needs resources, when your body needs fuel, it actually breaks down those proteins, those amino acids are released, and we use that for fuel. We use that for our reserves. So our muscle is a very important nutrient reserve. Hopefully none of you have starved for a prolonged period of time, but many of you have probably had, like, for instance, a cast on one of your arms. You broke an arm. 6 or 8 weeks later, when you take that cast off, are the size of your 2 arms the same anymore after that? You can use the yes or the no. Remember to respond to that. No, of course they're not the same size, right? And the reason for that is while you haven't been using those muscles, what happens is that your body's saying, all right, if you're not going to use those muscles, I'm going to break down those proteins in it and use them for other things in your body. So again, our muscle, one of the things we talk about is that use it or lose it. Muscles are absolutely one of those types of things. If you do not use the muscle, then your body's going to break down those things to be able to use it for something else instead. All right, questions on any of that? All right, I'll take the blinking of my screen to be that everybody understands. So we'll move on to the next characteristics. When we talked about epithelial tissues, we talked about the characteristics that all epithelial tissues shared. Epithelial tissues are superficial, they're polar, they're cell dense, they're avascular, right? Hopefully some of that stuff sounds familiar, especially since this is a cumulative exam, a cumulative class with a cumulative exam. Connective tissues, we talked about being deep, right? They have a matrix, they have fewer cells and more, substance in that matrix inside of it. We talked about characteristics that they all share. Now, muscle tissue, and I do mean all muscle tissues. And remind me again, how many different types of muscle tissues do we have? 3, that's right, excellent, perfect. We have 3 muscle types: smooth muscle, cardiac muscle, and skeletal muscle. All of those muscle types all share the exact same 4 properties. So all muscle tissues, the same way all epithelial tissues are superficial and cell dense, the same way all connective tissues have a matrix. These are characteristics or properties of all of our muscle tissues as well. So cardiac muscle, smooth muscle, skeletal muscle all have these characteristics. The first is that it is irritable. Right? If you're a Patriots fan, you're probably pretty irritable these days, right? With the loss of the almighty Brady, right? But what it means to be irritable, and again, when I think of irritable, I always think of my grandmother, because my grandmother was a loving, caring individual, except from noon to 1 in the middle of the day on Monday through Friday. Because from noon to 1, Monday through Friday, is when her stories were on TV. And if you talked during grandma's stories, she would throw her shoe at you. Right? And that's truly what it means to be irritable. To be irritable means that basically you can receive a stimulus and you respond to it. Right? Being irritable means you receive a stimulus and you respond to it. Right? Most parents stuck at home with their kids are becoming very irritable. Right? They're receiving a stimulus and they respond to it. Now, the way that muscle tissue responds is by producing an electrical signal. That electrical signal is called an action potential. Oh, I don't have that written there, so I'll go ahead and type that on here. That electrical signal that gets produced by a muscle cell when it gets stimulated is called an action potential. It is a big positive signal. And so it's irritable. It receives a stimulus and responds to it. And it responds by producing an action potential, a big electrical signal. So we can also say that it is electrically excitable, because it receives that stimulus and it responds to it. And let's move that over there to remind us of that. The second characteristic, and I want to make sure to make a point of emphasizing this, this is an active property. This is something that the muscle tissue actually does. Oh, great question. So again, someone just asked about the action potential. If the action potential is the same in muscles as it is in neurons. And the answer to that is yes and no. We do talk about muscle action potentials and we do talk about neuron action potentials. They're both big positive electrical signals. They're both stimulated in a similar way, but they're not actually identical. But yes, when we talk about action potentials, we have them both in the nervous system and we also have them in the muscle system, and they are going to be similar in properties. It's actually one of the reasons why we do the muscular system first, because while we're talking about the irritability and the excitability of muscle cells, that will help us to understand how we excite our neurons and how we stimulate action potentials in them as well. So this is kind of our baby steps into getting into this. All right, great question. Any others? All right, so like I said, contractility is an active property. That means it's going to use ATP, and this is something that actually the cell actually does. All right, and that is to contract. Muscle tissue has contractility. Basically, what that means, and so let's, hold on, text grab. Oh, I don't think I can do that. Hold on. So I'll have to put it up here. It's an active property. Uses ATP. Perfect. So contractility basically means that when it is stimulated, when it produces that electrical signal, that action potential, what it does is it contracts. It uses ATP, and that ATP is what shortens the muscle cell. That change in shape is what produces our force. It produces force. And that force, or we could think of it as, it's producing a force, it's generating tension. And that's how it does it work, right? This is the active property of how muscle cells do work. They are able to be stimulated, they generate an action potential, and as a result of it, they change their shape, they produce a force, they generate a tension. Now, muscle tissue also has two passive properties. One of those passive properties is extensibility. So again, this is a passive property. Right, this is not so obviously being passive, no ATP is used. Oops, no ATP is used in that. However, the other thing to remember is this is not something that the cell itself does. This is something that is done to the cell. What does it mean to have extensibility? Anyone know?
You can stretch.
Exactly, and absolutely, but there is a key to it. Extensibility means being stretched without damage, right? So you're absolutely correct. It's able to stretch, and it can do that stretch without being damaged. Now, it's like a rubber band that way. You can stretch a rubber band out without damaging it. Now, are there limits? Absolutely, right? If you can stretch it too far, and as a result of that, it could be damaged as a result of that. The other thing to think about is that is a passive property. If I take that rubber band and I just sit it there on the countertop and I stare at it for 4 straight days, is it ever going to be able to stretch itself out on its own? Right? It's not going to be able to do that. So it's something that can be done to the rubber band, a characteristic of the rubber band, but it is a passive property. It's not using any energy, and it's something that's done to them. And that's the exact same thing here, right? That extensibility is the ability to be stretched without damage. And then also, like a rubber band, when I stretch it out without damaging it and I let go, what happens to the rubber band?
It goes back to its original shape.
Absolutely, it goes back to its original shape. And the reason it goes back to its original shape is because it has elasticity. That elasticity is the ability to return to its normal length and shape. And so again, just like extensibility, this is a passive property. Oops. Right, no ATP is used, and it is something that is done to the cell. All right, so there you go. Those are the 4 characteristics that all muscle tissue, smooth muscle tissue, cardiac muscle tissue, skeletal muscle tissue all share. They are all irritable. They all have contractility, the ability to contract as an active property. And passively, they all have extensibility, where they can be stretched without damage, and they all have elasticity, is that where they will return to their normal shape and length. All right, questions on that? Irritability, that's a great question. So I was asked if irritability is passive or active. So again, you are welcome to ask questions either privately to me or to everyone. The advantage of everyone is everyone gets to see it, but I will always read them out as well, so that's fine as well. So let's answer the first question first. Is irritability a passive or active property? It is an that's a great question. Let's call it an active property, because producing an action potential is an active thing. It's a little bit of a cheat. It doesn't really use ATP while it does it, but it is definitely an active process that is taking place in the muscle cell. So it is definitely something that is active that way. While it doesn't directly use ATP, it does indirectly use ATP, and it is a process that is done to it. The action potential so it's both. So again, remember we talked about there's going to be a neural action potential and a muscle action potential. The neural action potential is where we actually make the decision and send a signal to the muscle cell. Then the muscle cell is stimulated. It's irritated, so we stimulate it. It responds to that stimulus by producing its own action potential. And that action potential is then going to be the trigger that is going to cause the muscle cell to contract. Right? If you think about when we've talked about some of the other processes, for instance, when we made bone. When we made bone, we had to change the environment. We had to change the chemicals. We had to change the signals. And that's basically what an action potential does. The muscle action potential changes the muscle cell's conditions so that it causes the muscle cell to go from relaxed to a contract state. And we'll actually go through that process, describing how that occurs. Great questions. Any others? All right, excellent. So now I get to clear the board. All righty. Now, as we've already hinted at, there are 3 types of muscle tissues, and so we need to talk about all of those. The first type of muscle tissue, move this down here out of my way, is skeletal muscle. Of course, skeletal muscle is called skeletal muscle, and here, of course, is the key word, because most skeletal muscle connects to bones. Of course, as we've talked about many times, what's the key word in that sentence?
Most.
Right, because most means not all. Absolutely. There's some of the best examples of this, for instance, are in your face. In your face, you have a ton of facial muscles that are all skeletal muscles. Many of them connect to bones, but others connect muscle to muscle, and some even connect muscle to skin, which is why we can be so expressive with our face, because we have tiny little muscles that actually connect to the skin instead of the bone and move our skin around so that we can look happy or sad or angry or confused or any of the other billions of emotions we can show with our face. So that is where it's located, and that is what it is called. One of the when we talk about some of the key characteristics, skeletal muscle cells are our biggest muscle cells. So they are the largest. They also, and here's where I get to take advantage of my amazing drawing skills. Oh, hey, I can cheat. Let's see if this works. Skeletal muscle cells are big, long, cylinder-shaped cells. They are our largest muscle cells. A single muscle cell can actually be up to 1 foot long. So for instance, in your leg, you have some single muscle cells that are over 1 foot long, and they have a long cylinder shape to them. There are some key characteristics I wonder if I can grab that now. Can I move that out of the way, or is that going to be a problem? I don't know how to move that. Oh, maybe select. Ah, cool. Put that up there. Hey, I'm learning. All right, excellent. When we talk about the characteristics of skeletal muscle, not only is it cylinder in its shape, not only is it the longest, but there's some other important characteristics as well. The first is that it is striated. Anyone know what striated means? Striped, absolutely. Striated means that it has stripes. So when we look at the cell, it has these striations. Oh, that's horrible. You get the idea. Pretend those are perpendicular to the muscle cell and parallel and all of these things. But what it has is it has stripes to it. And why that is important is actually because what those stripes are the precise arrangement of our proteins. One of the things that we're going to see in our muscle cells, but in particular in our skeletal muscle cell, is that the proteins that do the work are precisely arranged. And that precise arrangement allows for a precise motion, which allows for tremendous efficiency. Right? I could take any four of you and stick you in a boat, give you paddles, and you could get from one side of a lake to another. However, if I got a crew team and put them in the boat next to you and the two of you raced, the crew team would beat you every time. Not that you're not big, happy, healthy individuals, but that crew team has been practicing rowing, and more importantly, practicing rowing with precision. And their ability to all put their oars in the water at the same time, pull at the same time, lift their oars up at the same time, that precision produces more power, and that efficiency produces more power. So that precision and efficiency is going to be vitally important. And that's what we see in our skeletal muscles. The proteins are so precisely arranged that some areas have more, some areas have less. And so those denser areas look dark, the lesser areas look light, and so it actually gives the muscle a striped appearance. The second important characteristic is that it is voluntary. What that means is that we consciously control it. As you look down at your hand, if you want to flex your fingers into a fist, you have to make the decision to do that. And then you can do that, and you can relax it, and you can do it 3 quick times in a row, or not do it at all, or whatever it is, you have that conscious control. Of course, that conscious control starts in our nervous system, and so we get a brief introduction here to our nervous system. Our nervous system has different branches. And the part of the nervous system that controls our muscles, our skeletal muscle, is what is known as the somatic, because somatic, of course, means body, excuse me. However, we probably should sneak another word in here. It is our somatic motor, because it's what gives us our motor control. So it is controlled by our somatic motor nervous system, meaning that we have conscious control. And of course, as you also know, if I want to move my right hand, where do I make that decision?
The left side of your brain?
Left side. And if you remember way back when we were talking about directional and regional terms, we talked about that as being contralateral. So we have this contralateral control. Whereas if I want to move the left side of my body, I make that decision on the right side of my brain. If I want to move the right side of my body, I make that decision on the left side of my brain. So we have that contralateral control with our somatic motor nervous system. Now, the next characteristic of our skeletal muscle that you see here is that it is multinucleated. That, of course, means that it has more than one nucleus. What's interesting, normally when we draw a cell, we see the nucleus smack dab in the middle. But one of the things that's special about skeletal muscle, as we said, it has a massive number of proteins in it. So what ends up happening is we get all these nuclei, multiple nuclei, but none of them are going to be in the center of the cell. All of them are going to kind of be shoved to the periphery, shoved to the side of our muscle cell by all the proteins inside. So when we look at a skeletal muscle, we'll see that it has many nuclei, right? And they're all going to be on the periphery. They're all going to be on the outside. They're not in the middle because, like I said, this cell is chock filled with precisely arranged proteins. And a single muscle cell can actually have dozens of nuclei. Again, and if you think of what our nucleus contains, what does our nucleus contain again? Come on, these are the easy questions. What does our nucleus contain? Yeah, DNA. Absolutely, it contains that DNA. So again, this cell is chock filled with proteins. DNA is, of course, the instructions for making proteins. So we have lots of copies of the instruction manual that allow us to make all the proteins that fill this cell. Now, there is one downside to this incredibly elaborate, incredibly specific, incredibly precise structure. If you think about it, if you have, for instance, a skin cell that gets damaged, we have cells that divide to replace it. we kind of have something similar with our skeletal muscle cell. Our skeletal muscle cell, if you're working out because you have all this free time now because you're home and don't have to go to work and do all those other things, and you strain a muscle and damage one of the muscle cells, are you able to heal that and repair that? Absolutely. But what happens if a shark takes a big, huge chunk out of your arm? Can all of those cells regenerate back? No, typically not. So exactly what happens instead is, as we've talked about, instead it's going to heal via fibrosis. Instead of replacing it with muscle cells, we are going to replace it with a fibrous connective tissue, and that is going to limit its function because that fibrous connective tissue is not going to work the same way the muscle cells do. So skeletal muscle also, and I'll sneak this in over here as well, has limited regeneration. If there is minor damage to it, it's something that can be healed. But if we lose large portions of the muscle cells, it's not going to be able to replace those because these are such big and elaborate and fancy cells. It isn't just as easy as dividing one cell to form two. All right, so that is our skeletal muscle cell. And like I said, that's where we're going to spend a lot of our time. But as we also talked about, we have two other types of cells as well. The first is our smooth muscle tissue. Any idea where we would say our smooth muscle tissue is located?
In our digestion tract.
Say again?
Digestion, like our stomach.
Yeah, absolutely. That's a great example of a place we are finding. Again, we'll use that key word of most. Most smooth muscle is found in the walls of hollow organs. So that includes things like our stomach, our small intestine, all those other digestive organs, right? But also our blood vessels have smooth muscle in them. Pretty much any hollow organ in your body, with one big exception, is lined with smooth muscle in the walls of it. And again, that allows us to change the shape of it to help to move things through our body the way that we were talking about before. All right, but again, keyword there being most. One exception, for instance, one that we're all familiar with. What was the smooth muscle in our skin that made our hair stand up on end again?
Was it the erector pili?
Yeah, the erector pili muscle, absolutely. So notice we have seen some other examples where you have smooth muscle where it's not actually inside of a hollow organ. So there are going to be some exceptions, but most absolutely found in the walls of hollow organs. Excellent. Again, when we want to think of it in terms of its characteristics, and unfortunately this time I'm not going to be able to do the, oh, I can cheat. Excellent. Our smooth muscle, I'm going to sneak it into this space up here, has what we call a spindle shape. Spindle, oh, hope so I spell it right. Spindle shape. Spindle shape basically means that it has a large centrally located body and then two tapered ends. So notice I'm going to cheat and use my diamond shaped model here. So it's this kind of a shape where it has a large belly in the center, a big large belly in the center, and two tapered ends to it. So it's got one of the smallest shapes. If our skeletal muscle is the largest, our smooth muscle is the smallest with that spindle shape. It is going to have a single centrally located nucleus inside of it that way. And one of the big differences between our smooth muscle and our skeletal muscle, it is not striated. It does not have that same organization of proteins that our skeletal muscle does. This is going to be significant when we get to near the end of this section, because like I said, for the next couple of weeks, we are going to focus solely on how skeletal muscle contracts. And what allows it to contract are those precisely arranged proteins. So if our smooth muscle doesn't have those same precisely arranged proteins, then it's going to contract in a different fashion. And so, like I said, on the last day of lecture, we'll get a chance to go back and we will now come back to smooth muscle then and see, once we understand how skeletal muscles contract, how smooth muscle does it differently. Smooth muscle, as I also mentioned, is when we were talking about our skin, is involuntary, meaning that we don't consciously control it. That means it is a different branch of the nervous system that controls it. And instead it is our autonomic nervous system, right? Somatic motor is what controls our skeletal muscle, our autonomic nervous system, or again, we can, if we want to be fancy, put here in the fancy words, oops, there. And we can write in motor. So our autonomic motor nervous system is what controls our smooth muscle as well as hormones. So we have that as well. Now remember, we talked about how skeletal muscle has very limited regeneration. The anatomy of our smooth muscle is much more basic, much more simple. So of all of our three muscle types, this has the highest rate of regeneration. And I need to move that so that it's not on the motor. So our smooth muscle has the highest rate of regeneration of all of our muscle types. All right. So questions on our smooth muscle. Excellent. That brings us to our third type, our cardiac muscle tissue. And of course, where is our cardiac muscle tissue found?
The heart.
Absolutely. And more specifically, heart only. All right. One place and one place only. Whereas most skeletal muscle attaches to the bones, most smooth muscle is in the walls of hollow organs. You find cardiac muscle one place and one place only. And that one place we find it is the heart. Cardiac muscle is very, very special muscle tissue. And not surprisingly, it shares some of the characteristics with both smooth muscle and skeletal muscle. Let's talk first about one of its more unique characteristics, and I'll see if I can cheat and do this here. Cardiac muscle has what we call a branching shape. So when we talk about the shape of cardiac muscle, we say that it is branched, meaning that it has multiple parts that it can connect to. Instead of just a single shape, it has these kind of weird multi-limbed shapes to it. But it is going to share characteristics with both smooth muscle and cardiac muscle, and skeletal muscle. Like smooth muscle, it is uninucleated. It just has one centrally located nucleus. However, like skeletal muscle, our cardiac muscle is striated. So it is going to have stripes in the same way that our skeletal muscle does. That means it has that same arrangement of proteins, which means that it is going to contract in a very similar, notice I said similar, not identical, but it is going to use a similar mechanism to contract to what we're going to see in skeletal muscle. So that's going to be definitely beneficial for us that way. Again, so it's striated, it's uninucleated, and like smooth muscle, it is involuntary. However, there's something else very special about cardiac muscle. Cardiac muscle is also, and I'm going to put this in big caps to be able to emphasize it, it's autorhythmic. What it means to be autorhythmic is that it is able to generate its own heartbeat. It's able to contract on its own, right? If I cut your left hand off and put it on the counter, it's never going to contract on its own. Without that input from your nervous system, it cannot contract. If I rip your stomach out and I put it on the tabletop, again, even if I'm providing the cells with all the oxygen and all the nutrients and everything that they need to be living cells, without that input from the nervous system, it is not going to be able to contract. However, if I rip your heart out of your chest and put it on a beaker on the tabletop and I provide it with the oxygen and nutrients, it will happily contract and beat away. In fact, Your heart, if I ripped your heart out of your chest and I put it into that beaker on the countertop, it would beat at a rate of a little over 100 beats per minute. Now, by a quick show of yeses and nos on the participation, anybody here's heart rate going 100 beats per minute or higher right now? Yeah, unless you got a quad shot from Dutch Brothers before I started lecturing, right? Probably not the case, absolutely not. So notice what happens is our heart wants to beat really, really fast. However, we have the ability not just to necessarily control it, but the real key is we can modify it or influence it. So right now, the hormones in your body, and more importantly, again, your autonomic, and we can again, here, I'll cheat. See if I can grab, I don't know if I can copy these things. I don't care. Too much work. Again, our autonomic motor nervous system and our hormones can modify or influence it. It can make your heart beat faster or it can make your heart beat slower. Again, it's not something you voluntarily control. That can be a little bit tricky because if you think about it, if we all, instead of me sitting here lecturing, if we all closed our eyes and we put ourselves in the lotus position and we just sat here calmly and said to ourselves, our heart rate would go down. But is it because you're consciously telling your heart to go down? No, what you're doing is you're stimulating what we call the parasympathetic branch of your autonomic nervous system, which is your rest and digest process. So that would slow your heart rate down. The same way, if instead you closed your eyes and you envisioned a bear with an axe and coronavirus walking into your room, right, and you really tried to visualize what that would be like, right, a big bear with an axe coughing with a big, huge, dry cough, and if you really sat there for 5 minutes and thought about it, your heart rate would probably start to increase as well. And in that case, again, it's not because you're consciously telling it to do that. Instead, you are stimulating your autonomic nervous system, in that case, your sympathetic branch, that fight or flight branch, to get it to beat faster. So we can indirectly control it by the things we think about, but it's that autonomic nervous system, that involuntary control of our cardiac muscle. I've done an amazing job of drawing these cells for you here on this slide. And now, of course, I have to get rid of them all. But here we actually have some light microscopy where we can actually see some of these characteristics. And one of the nice things about doing this way is if in their classroom, if you're sitting way in the back of the class, you're not going to hopefully get to see it quite as nicely as we can see it here. So let's take a look at what we're looking at. Right now we are looking at some skeletal muscle. In the skeletal muscle, what I actually have here is 1, 2, 3, 4 pieces of cells. These are not full cells, it's just a portion. Remember, these are long cylinder-shaped cells. Notice we can see some of the nuclei, and notice as we talked about, this is the nucleus for this cell. We can see how it's shoved to the side because we can clearly and really nicely see the striations. The proteins here are so tremendously precise that we have a lighter region that we can see here and then a darker region that we see here. However, notice if you look closely, which since we're all sitting here at our computers, or at least your phones, hopefully you can be able to do, notice in the center of the light region, there is actually a small dark line. And if you look really closely, if you have a good resolution to your laptop or your monitor or whatever you're viewing this on, you should be able to see a slightly lighter region in the center of the dark strip. Can I get a quick yes or no as to if you guys can see that light line at the center of those? Excellent, perfect. So you guys are able to see that. Yeah, so again, these things are so precisely arranged that we have a light region with a dark strip in it, a dark region with a light strip in it, and that's because these proteins are so perfectly aligned. So we can see that. And again, to review the information, shape, they are cylinder shape. They are found, most of it, bound to or attached to bone, but not all of it. Yes, it is voluntary. Yes, it is striated. Many nuclei per cell versus 1 nuclei per cell. And it has a limited ability to be able to regenerate. So there we get a nice review of the characteristics of our skeletal muscle cell. All right. Here we have smooth muscle. Now, one of the things that clearly jumps out right away is we do not see the stripes. However, remember we said that this is supposed to be spindle shaped, right? Those spindle shape are these kind of elongated, big central body, tapered ends to them. But typically, when we see smooth muscle, it is arranged in a sheet like this. And the cells are interlocking, right? If you took your fingers, for instance, and you interlaced them with each other, they would all kind of come together into a single structure. And that's what we see here. We see a single layer or wall of smooth muscle. One of the things that we're going to get a chance to do histologically is to actually take some of these cells and pull this tissue apart. And when we do that, we'll see the spindle shape. So we'll have a chance to see that. So when they're typically in a wall like this, you can't see them. We know they're spindle shape. But the nice obvious thing is we don't see those precise stripes anymore. So again, when we talk about our smooth muscle, it has that spindle shape. And even though we can't really tell that by looking at this histology slide of it, we know smooth muscle has to be spindle shaped. We know that it is going to be found located within the walls of our hollow organs. It is voluntary. No, pardon me, it is not voluntary, right? We have that involuntary control of it. It is not striated, smooth. That's where it gets its name, smooth muscle, because it doesn't have the stripes. Only one nucleus, and it's located in the center of the cells. And as we talked about, it has the highest rate of regeneration. And then lastly, here is our cardiac muscle. Notice we see the branching shape. One of the things about the branching shape is there's a lot more space in between the cells. This space is filled with connective tissue. This connective tissue actually helps to form what we call a fibrous skeleton to your heart, which helps to give more structure to it. But when we look at the cells, we can see the cells here. They have, as we talked about, that nice branching shape. And we can clearly see the stripes. Again, this is a lower magnification than what we saw with the skeletal muscle, but if we got up close, we'd see darker and lighter regions within it. But here at the low magnification, we can clearly see some nice stripes. Cardiac muscle, of course, is found only in the heart. It is not involuntarily controlled. And again, remember, it is actually autorhythmic. It is striated, yes. It only has one nucleus. And notice this particular stain doesn't show the nuclei of the cardiac muscle cells. All these nuclei that we see are the connective tissue nuclei. So we don't actually see any of the nuclei in the cardiac muscle, but I promise you they're in there, one per cell. And here's the one question we didn't answer. What about cardiac muscle's ability to regenerate? Is cardiac muscle able to regenerate? No, it's not. Absolutely. Right. And that's one of the problems. Not only is cardiac muscle not able to regenerate, but it's also highly, highly dependent on oxygen. If a cell is deprived of oxygen for even a brief period of time, that cell can become damaged. And the problem is if that cell becomes damaged, we're not able to regenerate it. So again, our cardiac muscle, when damaged, right, that's a serious, serious condition because it's not able to regenerate. So cardiac muscle has no regeneration. Skeletal muscle has limited regeneration, and smooth muscle has the highest rate of regeneration. All righty. Any questions on that? All right. So as I mentioned, and let me go back. This is our introduction to the characteristics of all tissues, and now we've looked at all the tissues. However, from here to the very last lecture of this section now, what we are going to do is we are going to focus on skeletal muscle tissue, talking about its anatomy, talking about its organization, and building that up further. All right, I've talked for close to an hour, so I think this is a good chance to give everybody a chance to stretch, get something to eat, get something to drink, take a quick break. So what we will do is we will take a 5-minute break. Hold on, let me move this out of the way. So we will take a 5-minute break. I will pause the recording during that time, so you're not going to have to sit through it when you're going back and looking at this. My phone right now says 12:55, so that means we'll be back at 1 o'clock. And because I made this mistake on the in the 431 class, when I come back, I want to remember to start the recording again. All right, so when we come back at 1 o'clock, we will start the recording again, and we will continue now talking about and further talking about our discussion of our skeletal muscle. So I'm going to go ahead and pause this, and I will see you back here. Zoom in recording. And we can clear that. All right, before we hyper-focus on skeletal muscle tissue, any questions before we dive back into the material? Again, either raise your hand or text it onto the Zoom chat, group chat if you have any questions. All right. Excellent. A blank cursor tells me that we are okay, so we will move forward. All right. Our goal now is to understand how muscle tissue is made. Right? Again, as you look down at your arm and you see your big, huge, massive bicep brachia from all the exercise you've been getting for the past couple days, that is a muscle, that is an organ. The same way that our bones were organs, made up of primarily bone connective tissue, but other tissues as well. The same thing is true for our muscles. As you look down at the muscles of your body, they are organs. Now remember, in our skeletal system, we had 206 named bones. We, of course, have far more than 206 bones in the body, but 206 of them are named, and we had to learn them all. You have over 600 named muscles in the body. And as I'm sure you've already, over the past couple of days, looked at your anatomy list, you can see that you have far fewer than 600 that you are responsible for. But those are organs, and just like the bones, they're primarily made up of skeletal muscle tissue, but they're also made of other tissues as well. So when we talk about, oops, wrong button. When we talk about a muscle of your body, that organ of your body, it is made up of hundreds to thousands of muscle cells. One of the things that we will do, remember, skeletal muscle cells are long cylinder-shaped structures that can be up to a foot long. As such, they are also sometimes referred to as muscle fibers, and we can pretty much use those terms interchangeable. So if I say muscle cell or if I say muscle fiber, I mean the exact same thing. It's just that long, narrow, cylinder-shaped muscle cell or muscle fiber. Those mean the same things. So depending on which muscle we're talking about, whether it's one in your leg or one in your arms or one in your eyeball, outside your eyeball, it can be made up of hundreds or thousands of muscle cells. But those muscle cells are not just thrown together like inside of a bucket. They have to be precisely organized. And so to precisely organize those muscle cells so that they can function together, we need many layers of connective tissue. Oh, that's one of the things that I wanted to point out. I'm going to go back for a second. If you go back to, notice as we go back to our skeletal muscle cells, here we see the cells. Remember how we talked about, where's my spotlight? How precisely arranged the proteins are so that we have a light band and a dark stripe, light stripe and a dark stripe. But notice the dark stripe from one cell matches up with the dark stripe of the next, which matches up with the dark stripe of the next one, which matches up with the dark stripe of the next one. So not only are the proteins perfectly aligned within one muscle cell, but they're perfectly aligned between the muscle cells as well. And again, that type of precision comes by having connective tissues that hold and put everything into place. Now, when we look at the body as a whole, we have muscles, like the 600 named muscles of the body, that many of which work together. But just because two muscles work together doesn't mean that we don't want to keep them separate from each other. We don't want them to fuse together into one muscle. We want to keep them separate. And so our muscles are surrounded by layers of connective tissue. And the fancy name we give for a layer of connective tissue that surrounds and supports a single muscle organ is a fascia. So fascias are what basically help us to define and distinguish our muscles. Of course, that fascia is connective tissue, primarily collagen fibers. And you may not have thought of it in these terms before, but most of you, unless you're a very staunch vegan and have always been a very staunch vegan, are aware of this. right? Again, if we were in the classroom, I would love nothing more to get into the philosophical debate as to whether or not meat is murder, right? We can talk about that philosophically, but the one thing you cannot deny is that meat is muscle. So again, maybe not now because everybody's quarantined, but back in ancient times, you used to be able to go things like Renaissance fairs, right? And Renaissance fairs are great because you get to see people joust, you get to drink mead. But the best part of the Renaissance fair, besides the costumes, are of course the fact that you have the opportunity to grab a big, huge, massive turkey leg and walk around and eat it. And if you think about when you're eating that turkey leg at the Renaissance fair, or even doing it at Thanksgiving dinner, which is right around the corner, when we eat that, it isn't just a bone with a single smooth layer of meat wrapped around it, like a tube of meat on the outside. As you're biting into it, individual shaped muscles are being torn off. Because when you cook it in that temperature, you're roasting that chicken, that is breaking down that collagen, breaking down those connective tissues, and so the individual muscles come apart. So you're able to pull those individual muscles apart in that fashion. So we have that fascia that helps to distinguish them and keep them separate and distinct. There are different types of fascias. We know how our anatomists love their names. Way back in the integumentary system, we talked about the hypodermis. Remember, the hypodermis was not a part of the skin. It's that layer of areolar connective tissue and adipose connective tissue that is under the skin. We also called it the subcutaneous layer, so we use the term hypodermis and subcutaneous to describe it. Well, there's a third name for it. It's also referred to as the superficial fascia. The superficial fascia is basically, as we talked about in the integumentary system, what loosely connects our skin to our muscles. Right? Not only is Thanksgiving right around the corner, but Halloween is as well. I know none of these things are right around the corner, but you get the idea. On Halloween, if I were to go to the Hulk and I painted myself green, right, that would look awesome for the first 30 minutes or so. But as soon as that paint dried, every time I moved my body, that paint would crack. And the reason for that is when we contract our muscles change shape. In fact, that's where muscles get their name. Muscles come from the Greek word for mice, because as you look down at your forearm and you contract the muscles, you can see the muscles changing shape, and they thought it looked like little mice running around underneath your skin. So that ability for your muscles to change shape without tearing your skin is because we have that hypodermis, that subcutaneous layer, or now we can give it a third name, that superficial fascia that stabilizes it and holds it together. We also have a deep fascia. If you were there when we did our cadaver prosection, one of the things that I made a point of trying to emphasize is if you remember when we looked at the left leg of our cadaver, you couldn't see the muscles because there was a big, thick casing of connective tissue that wrapped around it. Part of our iliotibial band, when we get to legs, we'll talk about that, was in that as well. We called it the fascia lata. It stabilizes the muscles of the leg. It acts as a pressure cuff, and we talked about all those things when we did that. That would be an example of what we call a deep fascia. Deep fascias secure muscles that work together. So for instance, in your arms, you have muscles that flex your elbow. You have muscles that extend your elbow. And those muscle groups work against each other. So the groups that flex are bundled together in a connective tissue so they can work together. And the muscles that extend the elbow are wrapped together in a connective tissue so that they can function together as well. So our muscles, our individual muscles, those 600 muscles of the body, are stabilized by all sorts of layers of connective tissue to help them and support them in doing their job. Here, and again, I always like using the bicep brachia as the example, but if you look at the picture, you can clearly see that this is coming off of the femur. So it can't be the bicep brachia, but it is a muscle. And as we look at this here, we see that thing that would be the muscle, like the bicep brachia of your arm, a muscle in its entirety. And here is a single muscle cell. You guys can see my pointer when I do that, right? Quick show of hands or yes or no. Can you see that little highlighter thing when I'm done, when I'm using the spotlight? Okay,
Yes.
perfect. So this here is a single muscle cell. Notice this is a bundle of muscle cells. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and so on and so forth. I'm not going to bother counting them all. And then there's more bundles of muscle cells here, and they're all put together into a single structure that is called a muscle. So all these muscle cells, as we talked about, are combined and organized into the muscle by connective tissues. So it is these connective tissue layers that are going to protect and strengthen the muscle. Let me see something here. All right, so actually, I want to go from this to this. All right, I am going to now try to do some drawing for us. Again, you know how amazing my drawing skills are. They're even worse when I'm trying to do this with a stylus on my iPad. But I think it's still worthwhile to take the time to do this. So let's do this first with me drawing it so we can take a look at it and see how that goes. And then from there, we will go back to the pretty pictures and see this as well. All right, so give me a second. It takes me a moment to get this set up. Perfect. And then I got to make sure I have this oriented the right way. You guys able to see that line I just drew? And it's in the top left. Yes, 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 yes. Excellent. Perfect. All right. So let's go ahead and erase that. All right. What I'm going to do, the way the picture before showed, we looked from the big muscle and we worked our way down. What I'm going to do is work my way up. So what I'm going to start with is a single cylinder shaped muscle cell. That is a single muscle cell, a single shaped muscle cell. We know it's cylinder shaped, that muscle fiber. It has its striations, right? It has its nuclei on it, all those things that we had expected to see, right? And its buddy is sitting here right next to it. Now, one of the things we talked about is that muscle cells are irritable. And when they get irritated, they get excited and they produce an electrical signal. Yes, is there a question? I see a hand. All right, maybe I was just responding to that. You can see this. All right. So I have these muscle cells and they are electrically excitable. They're going to produce an action potential, right? You have wires in your wall that carry electrical signals as well, right? Fibers in there the same way this is a muscle fiber. And you don't necessarily want them to just be naked wires in the wall, because then the electrical signal could send from one to another and all sorts of havoc and other problems could be caused by that. So what we do is each individual muscle cell has a layer of connective tissue that wraps around it. So this one is individually shrink wrapped, and this one is individually shrink wrapped, and all of these muscle cells are individually shrink wrapped. They are shrink wrapped by an areolar connective tissue. Oops, ran out of space. An areolar connective tissue. Now, this certainly isn't the only areolar connective tissue we have in the body, so we need to give this one based on its location. And this areolar connective tissue that wraps around the individual muscle cells is what we call an endomysium, because it is within the muscle cells, within the muscle organ. So it is an endo. Endo means within. Mysium meaning muscle. So every single muscle cell within that muscle has a connective tissue that wraps around it. Now, these two muscle cells and 12 of its closest friends are going to be bundled together into one of those bundles that we saw. We, of course, need a name for this bundle, and the name for this bundle is a fascicle. And so our muscles have fascicles. Again, this isn't necessarily something you've thought of in these terms before, but I definitely know you're aware of this. Because again, as the weather was getting nice before we were all quarantined, maybe you decided to have a nice barbecue. So you went to Costco and got that nice big expensive tri-tip. You brought it home, you marinated it for 3 days. When it was finally time to cook it, you brought it up, brought it to room temperature, got that perfect sear on the outside, got that perfect pink color on the inside, brought it into the house and rested it for a full 30 minutes to get the juices absorbed back inside. And after all that loving care, time, and effort, did you just close your eyes and take your knife and just randomly cut it however you wanted to, just at all random angles? Anybody cut a tri-tip that way? No, how are you supposed to cut a tri-tip? Anyone know? No meat lovers out there? There you go. That's the magic term I was looking for, against the grain, right? Well, what the heck is the grain? Well, it turns out the grains are the fascicles, right? If you think about it, this fascicle, can you guys see the mouse on this when I do that, wave that? Cool, awesome. The fascicles are the grain of that muscle, of that meat. So what happens is, if you think about it, this is connective tissue fibers. So notice, if you cut your meat parallel to that, then you're going to have big, huge chunks of connective tissue, and it's going to be very, very tough. If instead you cut against the grain, you cut perpendicular to it, then what you get as a result of that is it breaks up the connective tissue, and it's a much softer, moister piece of meat as a result of that. So like I said, meat is muscle. And so we see that with the fascicles. So those fascicles are the bundles. That's the grain of the meat. And you see it in a lot of things. Or maybe you didn't want to get that really expensive tri-tip. So instead what you did is you bought that really cheap pork shoulder and you put it in the pot, in the crock pot, and you poured a beer on top of it and you put it in there for 12 hours. And after 12 hours, that meat just falls apart. But again, it doesn't fall apart into like individual cells. When it pulls apart, it shreds into these thin little wire-like threads. And those thin little wire-like threads are the fascicles. Because again, that slow, low cooking breaks down the connective tissue, and those fascicles just peel apart. So here we have these fascicles, and they are comprised of a dense, irregular connective tissue. So the fascicle is the structure. The connective tissue that forms that structure is a dense, irregular connective tissue. And of course, we have to give it a name based on its location. And because it is wrapping around the muscle cells to form the fascicle, we call it a perimysium. And so a muscle, like the bicep brachia in your arm, is going to be made up of dozens of fascicles. These fascicles, of course, have blood vessels that need to go to and from it. They have nerves that are going to control it. So all of those fascicles, plus the blood vessels and the nerves, are all going to be bundled together by yet another dense, irregular connective tissue. And this one is basically what brings all of these things together, and it forms our muscle, right? The organ. So that organ is formed by a dense, irregular connective tissue, right? This, remember, also is a dense, irregular connective tissue. So the ones that form the fascicles are a perimysium, the dense, irregular connective tissue that forms the entire muscle based on its location. We call it, because it's on top, we call it the epimysium. All right. All right. I've done an amazing job of drawing this. Again, it's one of the most beautiful drawings ever done in the history of everything. But let's go back to the pretty picture from the slide, and we can see this exact same arrangement again. Notice as we take a look here at the slide. Hold on. All right. As we look at the slide, and let's go back to my spotlight. Notice we'll work from our outside in. Here is that epimysium. That is that dense irregular connective tissue that forms the muscle. Inside of it are all of the fascicles. Notice here we've pulled out a single fascicle. This fascicle is also formed by a dense irregular connective tissue. This one we call a perimysium. But as you look inside that fascicle, we see a dozen or so muscle cells. But notice there's that little bit of connective tissue surrounding and isolating each one of the muscle cells. And that is our areolar connective tissue, and that is our endomysium. Here we see it in this picture in its entirety, but here they've got a little bit of a more simplistic illustration that shows this as well. Again, I started at the cell and built my way up. This illustration does the opposite. It starts big and breaks its way down. Notice here we have a cross section through a muscle, like the bicep brachia of your arm. It is the organ. Again, notice inside are bundles of muscle cells. Each bundle of muscle cells is known as a fascicle. So my participation thing's in the way, but I'll sneak 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So we have 12 fascicles in this single muscle. Notice there are blood vessels. The fascicle is an organizing structure, absolutely, right? Remember, this muscle is going to have blood vessels and nerves, and that's what we see here. And so the whole thing, the whole muscle is formed by this dense, irregular connective tissue that wraps around all the fascicles and all of the blood vessels and nerves. And that dense, irregular connective tissue we call the epimysium. All right. Now, again, these fascicles are the grain of the muscle, as we talked about. When we talk about muscle activity, we'll see that the fascicles kind of determine the direction that it is going to be moving the muscle as well. So that will help us to understand how that works as well when we get to there. So here is our epimysium forming our muscle. Notice they've highlighted one fascicle. So what they've done next is they've taken that one fascicle and pulled it out. Notice here, we see that fascicle has a dense, irregular connective tissue that holds all of the muscle cells in place. So here we have 1, 2, 3, 4, 5, 6, 7, 8 muscle cells all bundled together into one bundle. That bundle is called a fascicle, and it is formed by the dense, irregular connective tissue that we call the perimysium based on its location. Notice here in this fascicle, we can see that each muscle cell has its own connective tissue that wraps around it. Notice we also see all these dots inside the muscle cell. That's an indication that there's a whole lot of stuff going on inside this muscle cell that we haven't even gotten to and talking about yet. We got to break down that muscle cell first, but let's first pull out one muscle cell. And as we pull out that one muscle cell, we see that that muscle cell has all sorts of stuff like nuclei pushed to the side, like mitochondria that we recognize, and lots of proteins. That's what all these red dots indicate, are those are bunches of proteins. But what we see is that this single muscle cell has yet another connective tissue wrapping around it, and that is that endomysium. And that endomysium, remember, is an areolar connective tissue. All right. With me so far? All right, there's one more thing. I want to go back to my picture again and show you. So we'll do that. And then do that. Excellent. Now, the other thing to think about, and I guess I'll use, rather than going back to all the colors, I'll just cheat and use a different color. We'll use black. Areolar connective tissue has fibers in it, collagen fibers in particular, And those collagen fibers are going to extend beyond the muscle cell, right? The dense irregular connective tissue has collagen fibers that are going to expand beyond that as well. So again, these are all collagen fibers. So those fascicles are going to have collagen fibers that extend out from it. Our areolar connective tissue is going to have collagen fibers that extend out from it. And our epimysium, which is a dense irregular connective tissue, is going to have collagen fibers that expand out from it as well. So notice once the muscle cells stop, so even though the muscle cells have stopped here, all the collagen fibers of these connective tissues continue beyond the muscle cells. And notice what happens with those collagen fibers when they extend out. When they extend out, basically the collagen fibers all come together and become parallel. And when we have a bunch of parallel collagen fibers, that becomes our dense regular connective tissue. And what are one of the structures that a dense regular connective tissue can form? What do our dense regular connective tissues form? Yeah, see if I can give you a hint. Let's cheat and go back. Right? Dense regular connective tissues can expand beyond the muscle cells and be what attaches it to the bone. Exactly, forming our tendons. Now you're right, they can form ligaments as well, but notice in this case, we are actually going to attach those muscle cells to the bone, which is where most skeletal muscle is located, is attached to the bone. So all those connective tissue fibers of all of those epimysiums and perimysiums and epimysiums all come together to form the tendon that connects the muscle to the bone. And that's what we see in this last picture. Oh, actually, not in that last picture. We got one more picture that shows here. I'll cheat and come back to that. So again, here we have a single muscle cell pulled out. It is surrounded by an endomysium, which of course is an areolar connective tissue. It and 12 of its closest friends are bundled together into a fascicle by a dense irregular connective tissue that, based on its location, we call a perimysium. And here, this fascicle and 6 of its closest friends, along with some blood vessels and nerves, are bundled together by a dense irregular connective tissue to form an epimysium, and this forms our muscle. And then all those collagen fibers extend out from beyond the muscle cell to form our tendon to attach that muscle to the bone. We can see this on the illustrations, but the other place we can see this really nicely is when we look at this histologically. As we look at a histology slide of this, here we see a single fascicle. This single fascicle is again formed by a dense irregular connective tissue that, based on its location, we call a perimysium. But notice the muscle cells inside don't really touch. There's a little bit of connective tissue in between all of them. This tissue in between them is the areolar connective tissue. This areolar connective tissue is our endomysium. And this fascicle and that fascicle and the fascicle over here, as well as some blood vessels and some nerves, are all bundled together by a second layer of dense irregular connective tissue that is the epimysium. So epimysiums surround the perimysiums, which surround the endomysiums. Areolar connective tissue, dense irregular, dense irregular. All right. Questions on the organization of that? Again, if this doesn't make sense, it's just going to get worse. So make sure this makes sense, because if this doesn't make sense, like I said, it's just going to get worse. All right, excellent. Now remember also, as we talked about, a tendon connects muscle to bone. Most tendons are like this. They're narrow rope or band-shaped structures. But some tendons, like the one on top of our head or the one over our abdominal region, are big, broad, flat tendons And again, because our anatomists love to name things, our big, broad, flat tendons are known as an aponeurosis, IS being singular, aponeuroses being the plural. All right. Questions on that? Excellent. So now we have broken down a muscle, the organ that is a muscle, like your bicep brachia. We've seen how the muscle cells are organized. But as we have seen in some of the illustrations, right? So here again is our whole muscle cell, the organ. Here's our fascicle. Here's a single muscle cell. But as they've hinted at in that single muscle cell, there's a tremendous amount of anatomy going on in this single muscle cell as well. So what we need to do now is we need to break down a single muscle cell. To understand a single muscle cell, the first thing we have to understand is how you make one. Right? Back in ancient times, and by ancient times I mean when we were able to meet in the classroom, when we were talking about connective tissues, remember we talked about mesenchymal cells. And mesenchymal cells were the pluripotent stem cell. Oops. That are able to form all of our connective tissues. Well, here we have a new pluripotent stem cell, the mesendermal cells. These mesendermal cells are actually embryonic stem cells. These are stem cells that essentially aren't present as mature adults, although there's a little cheat to that. But it's these mesodermal cells that are actually used to make your muscle cells. Remember, when we talk about our skeletal muscle cells, one of the things we talked about is that they can have hundreds of nuclei. And the reason they can have, dozens upon dozens of nuclei is because they're actually not just made from one cell. Notice what happens is these immature mesodermal cells form myoblasts, cells that build muscle. And these myoblasts fuse together to form our single skeletal muscle cell. So when we have that single skeletal muscle cell, and I'll cheat and draw it there as nice and thin and skinny. I want to move it a little bit, move it down here. That single skeletal muscle cell is made up of hundreds of mesoblasts, mesodermal cells or myoblasts that have fused together to form it. When you have this type of a composite cell that is being formed, you can understand now when it's time to make new ones, where it can't just divide in half and those two cells can become totally new things. There's a tremendous precise arrangement to that. That also affects how they're going to grow. Right? Now that we have all this extra time on our hands, instead of studying for anatomy and physiology, we could spend 4 hours doing exercises, doing our bicep curls. And if we all did that for the next 8 weeks, when we came back, our arms would all be totally all jacked, right? We'd be yoked as a result of that. The reason for that isn't that you would make new muscle cells. What happens when your muscle cells get bigger or get stronger is that the muscle cells actually get bigger. What really determines your strength, and this is kind of a key point, the strength of a muscle is really due to the size of the cell. Yes, some of it is the number of cells, but it's really more due to the size of the cells. If we were to all just hang out and do bicep curls from now till the end of class, right? Arms day on Monday, Tuesday's an arm day, right? Rounded off with an arm day on Wednesday, and just for something unique on Thursday, we'd do more arms. As a result of that, the muscle cell would make more protein. And as it made more protein, what would happen is the muscle cell would get bigger. And when it gets bigger, it has more proteins. And when it has more proteins, it's stronger. Oops. Equals stronger. Right? That type of growth that we would talk about is what we actually call hypertrophy. when we use our muscles, as we talked about, use it or use it or lose it. When we use our muscle and we go in this direction, the more we use it, the more proteins we make, the stronger it gets. And that's the process we call hypertrophy. If, however, as we talked about, if you break a bone in your arm and you put that arm in a cast, then you're not using that muscle anymore. And remember, as we talked about, muscle is a nutrient reserve. If you're not using that muscle's not going to let all those proteins hang out inside that muscle cell and do nothing. Instead, what's going to happen is that your muscle cell is going to break down all that protein and our muscle gets smaller. And while you may not have heard the term hypertrophy before, but I'm sure you've heard what happens to your arm when you don't get to use it for a long period of time. It atrophies. So that atrophy is where it gets smaller, right? What happens is we get fewer proteins. It gets smaller, and as it gets smaller, it is going to be weaker. So a lot of the change in strength you see when people get bigger muscles. Now, again, when we're talking about these huge, massive, crazy looking bodybuilders, with that type of extreme increase in size, at that point, we will actually stimulate some cells to form new cells. And so in that case, we get a process we call hyperplasia. And you'll actually see that on the slide in just a minute. We're not talking about that. We're talking about normal working out, right? If you and I, we spent the next 8 weeks just working out instead of studying anatomy and physiology, your arms would get bigger, and that's because you were putting more proteins into your muscle cells, and your muscle cells were getting bigger. They were gaining hypertrophy. They were getting larger, and more proteins means that it is stronger. All right? So, like I said, with some of those bodybuilders in extreme conditions, so we go for hypertrophy, but in those bodybuilders, when we extremely stimulate those muscle cells, then what ends up happening is you can produce some new muscle cells. And remember, we did say that skeletal muscle had a limited ability to be able to regenerate. That limited regeneration comes from this cell right here. Notice as my myoblasts were fusing together, not all of them fuse to form that muscle cell. There are some cells that hang out right next to the muscle cell, and they stay immature myoblasts. Of course, those immature myoblasts have a special name, and that special name that we give for them is to call them satellite cells. So those myoblasts that hang out in there are our satellite cells. And so if this cell gets damaged, this myoblast can divide, produce more myoblasts, fuse together with this one to repair it, or replace it if it dies. But like I said, because we have a limited number of satellite cells, we have a limited ability to be able to regenerate. We can only regenerate the cells so much because we only have a few of those satellite cells, so it's limited regeneration. After that, it's just a fibrous connective tissue. All right, questions on that? All right, now that we know how to make a muscle cell, what we need to do now is actually identify the anatomy of the muscle cell. We know what cells look like. We've talked about cells before, but a muscle cell is a highly specialized muscle cell. So not only is it one way it's specialized, it has a special names to these things, but it also has some special structures inside of it as well. Now it is a little bit early, but we did take a shorter break before, and this is a good natural break point. We got a lot of content and a lot of material to cover. So I think this is a good point to take another quick 5-minute break, and then we'll come back and we will dissect this skeletal muscle cell and start breaking it down to its individual bits and pieces. But there's a lot of vocabulary that goes along with that. So I think that this is another good stopping point. So let's go ahead. We'll take another 5-minute break. I will pause the recording during this, and then of course remember to start the recording again. And let's see, my phone says 1:40. So let's come back at 1:45 and we will start back up then. So again, we can clear our heads real quickly, stretch our legs real quickly, and then come back and go deep into the anatomy and the vocabulary of this. So see you in 5 minutes. Right. Alrighty, so a couple good questions came in during the break, so let's start with those first. Mesodermal cells are indeed embryonic cells, so they are gone by the time we are born, but the satellite cells are not. Those are still immature stem cells. They're unipotent stem cells at that point that can divide to help to repair or replace damaged skeletal muscle cells. So mesodermal cells are indeed embryonic, but the satellite cells are not. The other question, which was a good question, is there is still going to be a lab. We don't really have a lab component of what we're going to be talking about today, but starting in the next lectures, we are going to be going over some of the lab stuff. You are responsible for the muscles, for the actions, for their origins and insertions, and I've got some things online that will help you. So I've made a note to try to make sure to go over it at the end of class. I posted on Canvas link to some materials that can help you for that, and then some of the other things we'll be doing. So I want to talk about that at the end of today's lecture today, but if I forget, please somebody remind me to do that, and we'll talk more about the lab component, because there definitely is a lab component. The good news is it is something that you can work on and do here at when you're at home, and study this pretty efficiently for the muscles, so it won't be too bad that way. All righty, any other questions before we dive back in? All right, so what we need to do now is talk about the anatomy of the muscle. And I think for this one, again, although it's a great picture here, I want to do a little drawing because I think it helps me anyway to make sure that I understand this stuff. All righty, so again, and the key here is we are looking at a single muscle cell. As we know, that single muscle cell is a big cylinder-shaped cell. And I'm going to cheat and open up the bottom of that. So we're saying we don't see the bottom of it. So we have this big, huge cylinder-shaped cell. Like all cells, it has a plasma membrane. But it is a very special plasma membrane. So I'm going to actually erase that. And instead, can I do text here? Ooh, I can. Well, I don't know how to type that. I'll cheat here. Let's do it this way. I'll leave that there and see if I can do this. If I put can you guys see that writing over the picture? Awesome, excellent. All right, so our oops, I don't know what I did there. There we go. Okay, excellent. All right, and so you see the sarcolemma right where the tag is. Do you see that the same way I do right here? Perfect. All right. So our plasma membrane is highly specialized. So we're not just going to call it a plasma membrane. We are going to call it a sarcolemma. And again, one of the things that we know is that our muscle cells are irritable. What makes the sarcolemma so special is that it is able to produce and propagate. And propagate is just a fancy word for send and action potential. So our plasma membrane of our muscle cell is specialized so that it can produce and propagate an action potential. So that's the sarcolemma. The inside is specialized as well. Normally we call what's inside the cytoplasm. That is the organelles, and that is the solution and the other things that are in here. But again, we are going to give it a fancy name, and in this case, we are going to call it the sarcoplasm. Oops. Plasm. There are a couple things that make the sarcoplasm special. Again, as we talked about, the sarcoplasm is what's special on the inside. One of the things that our muscle cells have is they have a large number of mitochondria. Mitochondria, of course, what produce all of our ATP, completely breaking down our glucose for being able to do that. So not surprisingly, we need a large amount of glucose. And of course, if you remember, animals like us store our glucose in the form of glycogen. Oops. So a massive amount of glycogen. So we have that fuel to be able to do that. But the other thing we need to break the glucose down efficiently in that mitochondria is oxygen. And so the other thing we need inside of our muscle cell to be able to store a large amount of oxygen is a molecule that can hold oxygen. Now, if you remember, we learned about hemoglobin, which is found in our red blood cells and holds oxygen. Well, it turns out inside of our muscle cells, we have a special protein, and that special protein is called myoglobin. And that myoglobin is what houses a large amount of oxygen. So what we have here is oxygen and sugar, and then the organelles to break that down and convert it into the ATP that this muscle cell needs. So here inside of our cytoplasm, oops, bring my picture back. Inside of our cytoplasm or sarcoplasm, we have a large amount of those three components. All right. Now, the other thing is, of course, we have organelles. And so there are going to be some specializations to all of this as well. One specialization goes back to our sarcolemma. With our sarcolemma, the sarcolemma doesn't just go along the surface of the cell. What actually happens is that there are invaginations of our sarcolemma that can penetrate deep down into the muscle cell. These are basically big tunnels that work their way down, and we call these tunnels the transverse tubules. And once you've written that out once on an essay question, you can abbreviate it as a T-tubule. But remember, the first time you have to write it out. Now, if you think about it, this transverse tubule is open to the outside. So that same interstitial fluid that surrounds this cell goes down into our transverse tubule as well. So this transverse tubule is going to be filled with interstitial fluid. Inside the cell, there are organelles. One of the biggest organelles is all of those proteins. As we've talked about, we have these massive, massive collections of precisely arranged proteins. And this structure runs the entire length of the muscle cell. And this structure is known as a myofibril. Actually, let's not do it that way. Let's do it this way. We have this myofibril. The myofibril is our contractile organelle. So basically, it is the structure that is responsible for producing our contraction. It's what generates the force. Put it there so it's out of my way. So that myofibril is the contractile organelle, and it's what's made up of massive amounts of proteins. These proteins, oops, wrong button, are precisely arranged into a repetitive pattern of structures that are known as a sarcomere. Oh, and again, I'll cheat and write it out. These sarcomeres, these are the structural and functional units. All right. So again, structural, as we talked about, one of the characteristics of muscle cells is that they have stripes. It is the precise arrangement of the sarcomere that gives us our stripes. And it's the functional unit. This is what is generating the force. This is what is generating the tension. This is what allows the muscle to change shape so that it can do its work. Now, I'm going to change colors here. Or at least I'm going to try to. Oh, there we go. Our myofibril is all these proteins. But, and I'm going to kind of show it as I'm going to first draw it around the outside like this. But what we need to know is that it has this kind of lace-like structure that goes around the entire myofibril, wraps around the myofibril. This structure here is the smooth endoplasmic reticulum. Remember, with our smooth endoplasmic reticulum, we talked about sometimes it can be specialized, and that specialized sarcoplasmic reticulum can be used for storage. In this case, its job is going to be to store calcium. And this special smooth endoplasmic reticulum we call the sarcoplasmic reticulum. And like I said, its job is to store calcium. Oops. Uh-oh. Give me that back. There we go. So we have our sarcoplasmic reticulum here that stores calcium. But there's something else interesting about the sarcoplasmic reticulum. The ends of it are enlarged. Right where it meets the transverse tubule, it becomes a big enlarged structure. And that big enlarged structure is what we call the terminal cistern. Cisterna or cisterna, singular, plural would be cisternae. So we have this enlargement of it there. Now, this happens on the other side as well. There is another terminal cisterna here and another sarcoplasmic reticulum there. And so notice what we have in this area, and I'll change colors again. Two enlarged terminal cisternae and the one transverse tubule together, we give them a fancy name, and those three things together we call the triad. Because after all, oops, so I spell it right. Oops, wrong button. We call that the triad because it's three things. 1, 2, 3. Two terminal cisternae and the transverse tubule, collectively we call the triad. Now, again, this picture that I've drawn with all of its labels is incredibly messy. But if we look at the pretty picture from the textbook, we can see all of these same structures as well. So now that I've done a hideous job of drawing this, let's switch back to our lecture where we can see these pictures and see these things a little bit nicer. So let's go back to sharing the outline. Share that. Perfect. And now we can go through these things a little bit nicer. So again, as we talked about, the plasma membrane, and let's go ahead and use my highlighter. Plasma membrane, the outer surface, is the sarcolemma. It is a special plasma membrane, and as we said, it is specialized in that it is capable of producing and propagating, which again is just a fancy way of saying spreading. It can produce and spread an action potential. Notice that a plasma membrane folds down to form these tunnels that penetrate deep into our muscle cell, and these are our transverse tubules. So here we see those transverse tubules in yellow, which are those extensions of the sarcolemma. The inside, which is the organelles and the fluid components that are in there, are our cytoplasm, but again, our cytoplasm is specialized, so we give it a special name. It is the sarcoplasm. And as we said, three of the things that make the sarcoplasm so special is that it has a massive amount of glucose, the fuel, the energy, right? If you're going to drive that car, you need a big gas tank. So this is what we have here. This is our gas tank. This is our fuel. We need the mitochondria, which is the engine that makes the ATP so that we can do the work. And again, to completely break down that glucose to make our ATP, we need oxygen. So we have a large amount of myoglobin in here as well. This is what we're going to use to make our ATP so that this muscle cell can work. As I mentioned, we have those invaginations of the sarcolemma that penetrate all the way down to the depths of the muscle cell. Notice they wrap around the organelles on the way there. And those are those transverse tubules, which again, once you spelled it out once, we can call T-tubules. They tunnel towards the center of the cell, and they contain interstitial fluid. Notice we can see some of the openings to them up here and here and here. Here in blue, we have our sarcoplasmic reticulum. Remember, this is our specialized smooth endoplasmic reticulum. Notice it wraps around our myofibrils. The myofibrils are those contractile organelles we talked about. And it stores calcium. As we've talked about, calcium makes cells do wonky things. Right? And in this case, the wonky thing our cell is going to do is going to be to contract. So here in our sarcoplasmic reticulum, we have that calcium to do those wonky things. That is our sarcoplasmic reticulum. And notice at the end, it gets really large. That enlargement at the end is called the terminal cisterna. And notice there's a terminal cisterna on this side, a terminal cisterna on this side, and the transverse tubule in between. And so these three things together we call the triad. All right. However, as we also mentioned, the most unique organelle of our skeletal muscle cell, and here we see one pulled out as well, is our myofibrils. Myofibrils are our contractile organelles. They run the entire length of the muscle cell, and they are made up of massive numbers of proteins. There are literally dozens of different proteins that work to form this so that they have their precise structure, so they produce the force and do everything else that we want them and need them to be able to do. Those proteins are what are known as the myofilaments, right? They're protein filaments in the muscle, so myofilaments. And some of them are smaller and some of them are larger, so they are often known as the thick and thin filaments. And we'll talk about which ones are the thick ones, or which one is the thick one. And pretty much all the other ones are thin ones that we'll see. What you also see a hint of, and you see it nicely here in the center of the model, is that what we have is this repetitive pattern. Notice if you look, there's this pattern of this jagged edge here, and these red and blue proteins, and then that jagged edge here, and these red and blue proteins, and a jagged edge here, and there'll be more. It's kind of like if you've ever been stopped at a train as a train crossing is going by, and that train goes by, and it's like a transport train. So you have just hundreds of grain cars, just a repetitive pattern of grain car, grain car, grain car, grain car. That's what we have. We have this repetitive pattern of the sarcomeres. And as I mentioned, this sarcomere, this structure right here, which now we're going to have to pull out this myofibril and look at it up close and personal. This is both the structural and the functional unit. The sarcomere is this precise arrangement of proteins that gives the muscle its stripes, but it's also the functional unit. This is what contracts to produce the force. Here, we've pulled out a chunk of a myofibril, and here we actually see many of the proteins that are in this. Now, as I said, there are literally dozens, plural, of these myofilaments. I'm only going to hold you responsible for 7 of them. So of the literally dozens that are found in here, there's 7 important myofilaments that we need to be able to identify and see. And between here in the illustration and here using an electron microscope to actually look at a myofibril in a muscle cell, this is actually a microscopic view using an electron microscope of this organelle inside of a muscle cell. And from here to here is our sarcomere. We will talk about and identify 7 of these proteins that you are going to be responsible for. All right. How do I want to do this? Again, normally this would be easier. Yes, so it is. So great question. Let me go back. Absolutely. And we kind of get a hint of it here. The precise arrangement of these myofilaments, where the proteins are, which proteins are located in which location. Notice when we look at it in an electron microscope, there's some areas that have much denser proteins and some areas that have much lighter proteins. And it is this precise arrangement of the proteins that actually gives the muscle cell its stripes. So absolutely. So this is where we get the stripes. This part here is the light stripe of the cell. This is the dark, and then another light, and there you see another dark. And so, yeah, that repetitive pattern that we have, those stripes come from the arrangement of these proteins. All right, so here's how we're going to do this. What I want to focus on first, I'm not going to worry about the names of them. We'll come back to this, but I do want, there are 3 main types of muscle proteins. So myofilaments that we're going to see. The first are the contractile ones. The contractile proteins are the ones that generate the force. They're the ones that generate the tension. The second are our regulatory proteins. Again, every single muscle in your body has the ability to generate force, has the ability to contract. But is every single muscle in your body contracting right now? No, of course not. And the reason it's not is because we have the ability to turn those muscles on and off. It turns out the contractile proteins want to interact all the time. If you let them, they would constantly contract and your muscles would be constantly producing tension. And that would be a bad thing. So what we need are regulatory proteins. And those regulatory proteins can, as the name indicates, regulate. They're going to turn them on and off. They're going to determine when they can interact and when they cannot interact. And when they interact, not only are they going to produce force, but they're going to produce that force. We want it to be efficient in that. We want that precision, and that precision gives us our stripes, the structure, but also is what generates and maximizes our force. So the third type of protein we need are structural proteins. So there are 3 main types of myofilaments. Contractile, those are the ones that generate the force. Regulatory, those are the ones that turn it on and off. And structural, which are the ones that give it its shape. All right. With that, we can go back to our drawing now, and I can draw this, and then we'll look at a pretty picture to see how this is going to work. So let me again go back to the iPad. Perfect. All right, we need a new picture. How did I rotate it again? There we go. Clockwise. Perfect. All right. So, what we're going to start first with are the contractile proteins. So again, what we are looking at now, we are looking at a single sarcomere. And the proteins that are involved in this. Let's start first by talking about the contractile proteins. I know I can do this. And there are two. Those two contractile proteins are myosin and actin. Actin is a dimer. By dimer, what I mean is that it's made of two subunits. Basically, it is two ball-shaped subunits, and that right there is basically what an actin looks like. It's very simple and it's very basic, but the nice thing about it is that it's easy to put them together. I can put them together and I can form a big long line of actin. And basically it forms a long fiber-like structure, right, our filament. Myosin is our motor. Remember, as we talked about, motors convert chemical energy, oops, not how you spell convert, into mechanical. And they do that by basically having a specialized head-like structure. I'm going to cheat and put this up here. So our myosin basically has a long tail-like structure. And then it has a head region. It is the head that is the motor. Basically what it does is it uses ATP. And when it uses ATP, it basically changes its shape. It moves. So it rotates. Now myosin, and that's where I can come over here and switch back and forth between these two drawings. Our myosin forms what we call our thick filament. And I think I need to extend that out a little bit more. All of the tails, hundreds of these tails are put together in a big, thick structure. And then what happens is all of the heads of that myosin stick off of it. So we have all these myosin heads. That come off of it. And excuse me, what that myosin head wants to do is it wants to grab onto the actin and then it wants to pull. It wants to pivot. Right? It basically grabs and pulls. Now you could be grabbing and pulling right now and you're not going anywhere because you're not grabbing and pulling on anything. So what our myosin heads need to grab and hold onto is that actin. So remember, actin is a dimer. So there's all these myosins, all these actin dimers that are like this in a row. And I'm going to cheat for the rest of them and just draw them as lines. So these lines that I'm drawing are my actin. But remember, it is a dimer. And on that actin, there are binding sites. All of these actins have a binding site. That the myosin head can grab and pull on. Okay? So those are our two contractile proteins, myosin and actin. The actin is something that needs to be grabbed onto by the myosin head, and then the myosin head is going to use that motor to pull on it, and that is going to pull, and that is going to generate our tension, and that is going to generate our force. Now, if there was just a rope in the middle of your floor, and you picked up an end on it, and you pulled and tugged on it, again, you wouldn't go anywhere. That other end of the rope has to be anchored to something. So you could either tie it to the wall, in which case you grabbed and pull, and you would start moving towards the wall. Or probably a better example would be to tie it to your end table. If you tied that rope to your end table and then you started pulling on that rope, the end table would start moving towards you. And that's what happens here. What our actin is connected to is to our first structural protein. Our first structural protein is this zigzagged protein known as the Z disc. So if we go back to our previous, we can now start talking about our structural proteins. And our first structural protein is the Z disc. The Z disc basically is the boundary of our sarcomere. It is the wall, right? I have a wall right here next to me, and that wall right here next to me not only is the end of my room, but it's also the beginning of the room next to mine, or at my daughter's room. And so it's the same thing here. The same way that there is actin coming off of this side, so that we have a sarcomere here, there's also another sarcomere going off that side. So there's more actin going off on this side and more actin going off on that side. So this is a sarcomere, and this is a sarcomere, and this is a sarcomere. And again, we have that repetitive pattern of the sarcomeres that are going on. And I need to cheat and move that S. So we have those sarcomeres that are being formed. So the Z disc is what our actin is attached to. Let's make sure all of our actin is attached. And again, this one attaches as well to that. So when the myosin head grabs and pulls, it's going to grab the actin and pull. Then what happens as a result of that, as it grabs and pulls, is the Z disc gets pulled as well. All right. Now, notice the actin is kind of stabilized in place, but our myosin head isn't. So we need a protein to help the myosin head as well. That protein that helps to stabilize the myosin is this big, long, coiled protein. And this big, long, coiled protein is called titin. Titin is named after the Titans. Remember in Greek mythology, the Titans, like Atlas, are who ruled the world before the Greek gods did. And the Titans were massive creatures. Well, at the time this protein was found, this protein was the largest protein that had been found in the human body. And so they called it titin based off the fact that it was so large. There are two important things about the titin molecule. Notice the first is that it helps to stabilize and hold the myosin filament in place. But the second part of it is this coiled end. This coiled end, even though I haven't done a very good job of drawing it, is like a slinky. And if I have a slinky and I hold the two ends of the slinky, I can stretch that slinky out. And do I damage it when I do that? No, it's not going to exactly. Thank you, Jesse. No, it can be stretched. It has elasticity. And this is where the elasticity of our muscle cell comes from. And when I let go of that slinky, it goes back to its original shape. And so the elasticity, so the extensibility and elasticity of our muscle cell comes from this titin protein. Now, yes, to answer the question that was asked, the Z disc forms the Z line. So the same way that a table is made of wood, the Z line is made of the Z disc. So yes, so you can kind of use those terms interchangeably. Notice I've only drawn one titin. There is a second titin molecule that's on this side as well, and it goes across and meets there. But they don't quite meet in the center. Well, that's weird. That didn't show up on my picture. Why didn't that show up on my picture? What's going on? All right, hold on. All right, now they're there. Perfect. Okay. I don't know if you guys were able to see that before, but I couldn't. So there's titin on both sides. So the titin from both sides come together towards the middle, and they meet towards the middle of the myosin. Now, this one myosin isn't just going to hang out by itself. There is another one in the adjacent portion of the myofibril over here, and dozens more as well. So we need a third structural protein to hold these things together. And that third structural protein basically is not just one, not just two, but 3 lines of interlocking proteins right down the center, that these proteins are Myoglobin. So if we go back here, our third structural protein is Myoglobin. There we go. And it is a stabilizing protein. Notice, if you remember, when we looked at our muscle cell histologically, all of the dark lines, all of the white lines were all perfectly arranged, and they kind of matched up with the cell next to them as well. That's because structural proteins like this Myoglobin will actually connect the not just these individual myosins together, but they connect all the way to the plasma membrane of the muscle cell and can actually cross to the next cell as well, stabilizing them all together. So 2 contractile proteins, 3 structural proteins. The only thing we have left then are our regulatory proteins. And there are 2 of those. Our structural proteins are what hold everything together and give it its shape. The contractile proteins are what generate our force. But as we talked about, that myosin always wants to hold on to, grab, and pull on the actin. And if it was doing that, that would cause the muscle to contract. That's weird. Sorry, hold on. I've lost my chat. I've lost my participant list. And this is a new process for me too, so we're hopefully figuring it all out. Excellent. All right. Okay, got those back now. So again, we have our structural proteins holding everything in place. We have our contractile proteins that want to grab and pull, generating that tension. So what we need is a way to regulate them. And to do that, we need 2 regulatory proteins. And the first regulatory protein is called tropomyosin. Tropomyosin is a long thread-like protein that basically sits between the myosin and the actin, basically stopping the myosin heads from being able to grab onto the actin. So this protein here is our tropomyosin. It basically is a big wall, big thread-like structure that sits on top of the binding sites so that myosin can't grab. And as myosin can't grab, the muscle is going to be relaxed. And that's great. Now our muscle is not going to contract just willy-nilly or be contracting all the time. But we do eventually want that muscle to contract. And so when we want that muscle to contract, we have to use our second regulatory protein, and that is troponin. Troponin is basically a handle-like protein that sits on top of the tropomyosin. So this is our troponin. And its job is, when it's time, it is going to take that tropomyosin and move it out of the way. And so when it moves it out of the way, now the myosin can grab onto the actin. Now it can start the contraction, and our muscle is going to generate its force. All right, now, like I said, I appreciate that these aren't the best drawings that have ever been done in the history of mankind. So let's take a look at our pretty pictures from our slides to see if we can make some sense of it there as well. Here we see our myosin, our thick filament. Again, notice here, where are my annotations? There we go, spotlight. So again, here we see a single myosin. Notice it's also a dimer. Here's the head region, here's the tail region. And notice hundreds of these tails are put together so that those myosin heads stick out, and that is our thick filament. As we talked about, that myosin is a motor. So here is that head, and what it does is it uses the energy of ATP to basically pivot. Right? It's a motor. It converts chemical energy, in this case ATP, into a mechanical energy, into the force of that contraction. Here we see that dimer, and again here we see just the individual subunits of our actin filament. They're the ones in blue. And notice they have that handrail. For the myosin head to grab and pull, the myosin head has to have something to grab onto. So these are what are called the active site, or what we also sometimes refer to as the binding site. So this is the handhold that the myosin head can grab and pull. So here we have that long line, and again, that is going to be attached to our Z disc. And notice here we also see our regulatory proteins. Here's that long thread-like tropomyosin. Notice it's blocking all of the binding sites, so that myosin head cannot grab it. But luckily there's a handle here. This handle is the troponin. And something is going to cause this troponin to change shape and move out of the way. And when that something causes the troponin to move shape and change shape and move out of the way, then myosin can grab. Any idea what might change the shape of troponin? Getting that muscle to contract is definitely a wonky thing. So what kind of wonky thing do you think could change the shape of that troponin and move it out of the way?
Calcium.
Absolutely, calcium. Calcium is what's going to bind to the troponin, change the shape of the troponin, and move it out of the way. And that's how our regulatory proteins work. I love this picture from your textbook. Here is, again, they use this electron microscope to show us these microscopic views of the proteins inside of our cells. Here we have our big, thick myosin filament. So here's our myosin filament, and we can see the heads sticking off of it. Notice those heads are binding to our actin. Here's the actin, which is that thin filament. And notice, again, it's a repetitive pattern. On my drawing, I just had one. We just basically looked at this much of it, right? Or let's do a drawing. We were just looking at this much of it in our, the color of that is horrible, but that's all right. There we go. Just that much of it. But again, there are dozens of these in here, each of these myosin heads capable of pulling and generating force. So we have this massive repetitive pattern of myosin and then actin and myosin and then actin and myosin and then actin again and again and again. All right. Here we get a better view of those regulatory proteins. Again, we have the tropomyosin, which covers the binding site, stopping the myosin from getting to it. And then we have that handle like troponin, which is going to be able to change its shape, move the tropomyosin out of the way. And calcium is what's going to be the magical thing that's going to allow that to occur. So those are our regulatory proteins. And then here we see our structural proteins. Again, it starts first with our Z discs. Here we see the Z discs. They are forming the proteins that are forming the boundaries of this sarcomere. But notice also they're the boundary of the next sarcomere that would go on that way, and the boundary of the next sarcomere that goes off that way as well. Here we have the titin. The titin, again, notice it's got this big, huge coiled portion. This is what gives it its extensibility, allows it to be stretched without damage. And this is also what allows it to recoil, have its elasticity. And then it also is going to help to stabilize the myosin. Notice the actin binds to the Z disc, but the myosin doesn't. So the titin helps to stabilize it, one from one side and one from another. And then here we have those three interlocking proteins that are the myomysin. And that myomysin stabilizes not just these, but like I said, it connects all the way to the plasma membrane of the muscle cell and can expand beyond that as well. So while there are dozens upon dozens of proteins inside of a myofibril, these are the 7 that are going to be most important for us. These are the 7 that we need to know and understand and that you're going to be responsible for on the exam. All right, questions on that? All right, so as I mentioned, the Z disc is our structural unit and our functional unit. Functional unit meaning it's going to be the interaction of these structures, the movement of these structures that generate the force, generate the tension. But structurally meaning that this is what gives it its stripes, right? So when we talk about a Z disc, when we talk about, pardon me, a sarcomere, it goes from Z disc to Z disc. But we also have our stripes. There is a dark region. As we mentioned, the dark region is the part that is dense with proteins. And the densest protein structure is the myosin. So notice this portion of our sarcomere that contains the myosin is the part that is going to form what we call the dark band. The part that doesn't contain myosin, which basically goes from the end of this myosin to the beginning of the next one, this region in between is what we call the light band. And so those are the names for them, dark band or light band. There are 2 other names for them as well, A band and I band. Now, the term A band and I band actually come from the German words because it was a German anatomist who first described this, so they're German words for this. But what I find really helpful and helps me to remember is the dark band, which has A in it, is the A band. The light band, which has I in it, is the I band. Again, I emphasize that is not why they have those names, but it is a super easy way to remember which is which. Light with the I is the I band, dark with the A is the A band. All right, I'm gonna jump to the next screen. Oh, okay, so I lied. Okay, all right. So we have our sarcomere, we have our A band and our I band, but there are 2 other regions in here we need to be able to identify as well. The first is what is known as the H zone. Notice for most of the A band, again, the A band is the part that has myosin. For most of it, actin and myosin overlap. However, notice right here in the middle is a small portion where there is no actin, where it's just myosin. This small part right in the middle is what is known as the H zone. However, remember, we also have that myomysium, that protein that stabilizes all the myosin. It forms a dark part right at the center, and that dark part right at the center is what we call the M line. So A band, I band, H zone, and M line, as well as Z line, which is formed by the Z disc, are all features that we can see on this. One way to think about how these areas are defined are by the proteins that you find in those areas. Notice, I love this picture from your textbook because it does a nice job of showing this. Notice if we were to cut our myofibril in the I band region, H zone is the part that does not have, just has myosin, does not have actin. And we'll see that here with a pretty picture. So if we were to cut this myofibril right here on the in the I band, notice in this I band you would only have actin. Yes, there'd be titin as well, but you wouldn't see it. We would just see the actin. If instead we cut it in the A band, notice we would see the actin and the myosin overlapping. However, and this will answer your question, if we cut it in the H zone, notice the H zone would just have myosin. It would be this area here without any actin in it. That would be the H zone. And if we cut it right on the M line, there still would not be actin. There would be myosin, but what we would see is the myosin would be interconnected, and that's interconnected by that myomesin proteins. So here is that myomesin protein. So here we can see those distinct regions by what proteins would be if we cut it. If we cut it on the M line, we see myomesin and myosin. If we cut it in the H zone, we just see myosin. A band, we see myosin and actin. I band, we just see actin. This illustration does a decent job of showing this for us. Here, things are stretched out a little bit more. Notice we don't have regulatory proteins, but that's okay. They don't affect the colors. A band is the area that contains myosin. In that A band, actin and myosin overlap. But at the center of our A band, we have our H zone or H band, the area that just has myosin. And at the very center of our A band, we have our M line. Notice one other thing, our A band is completely contained within a single sarcomere. But our I band starts in one sarcomere, crosses the Z line, and goes to the next one. So I bands are not contained within one sarcomere, whereas the A band is. Again, we can see this nicely on the illustrations. But as we also pointed out, your book has these great electron microscope views. This is, using an electron microscope, the actual myofibril portion of a muscle cell, and we can see here. Notice here we can see the boundaries of our myofibril. This would be like where the transverse tubule and the sarcoplasmic reticulums are wrapping around the outsides of them. This dark line here is the Z line formed by the Z disc. So again, from this Z line to this Z line is a single sarcomere. In the center of that sarcomere is the area that contains myosin. And this is the A band, the dark band, completely contained within one sarcomere. But from the end of one myosin to the beginning of the next myosin is the area that does not contain myosin, and that is our I band, the light band. Notice at the center of that I band is that dark line. We see the Z disc formed in the Z line. Notice also at the center of our A band, we have an area that is slightly lighter in color. That is the H band or H zone. Those terms can be used interchangeably, where it's just myosin. And notice it's a little darker just at the center. That's where the myomesin is, and that would be the M line. This picture is just from an electron microscope. Here, whoops, wrong button. This one, it's actually been colorized. So it's been colorized, and we see the same thing. Now, I do want to point one thing out. Notice that my myofibrils are not perfectly aligned. Here's this Z disc, and here's this one, here's this one, and here's this one. But you have to remember how they get these electron microscopy views. First, they have to take a chunk of muscle out of something, right? Then they freeze it. Then they slice it tiny. They put it on a slide. They put it in this electron microscope, and then they bombard it with electrons. The tissue is actually destroyed in the process. So not surprisingly, with all that processing, that it's not surprising that things won't be lined up as perfectly as we're freezing it and cutting it and staining it and doing all these things to it. But it's still pretty close. So here's one myofibril, here's the second. Here is our sarcomere from Z disc to Z disc. Here is our I band, which crosses the Z disc. Here is our A band that contains the myosin. Here is our H zone, and right at the center is the M line. And if you think about it, whoops, this is exactly what we saw here. Notice when we first looked at our skeletal muscle, this is our A band, the dark band. But remember, we see that slightly lighter stripe down the center. That's the H zone. That's the area that just has myosin. Now, at this magnification, we're not going to see the M line. So we're not going to see the M line, but we can see the H zone. Here's our A band. And notice, as we saw at the center of our I band, pardon me, the light band, the center of our I band, we have that Z disc. We have that Z line. So there's sarcomere from Z disc to Z disc, or Z line to Z line. We have our A band, our I band, our A band, our I band, our Z line, and our H zone. We can see all of that when we were looking at it this way here. All righty. Questions on that? I know that is a lot of anatomy, a lot of vocabulary that we have to learn, but to understand how the muscle contracts, we have to understand how each of these individual proteins are going to work and how they're going to interact with each other. We're going to need to do that. Now that we've broken it down to its most basic components, the actual proteins that are making our muscle, what we can do now is start building it back up. First thing we are going to do is see how these proteins interact with each other. And once we see how these proteins interact with each other, we can put them back together into a sarcomere. We can then put that sarcomere into a myofibril. We can put that myofibril into the muscle cell. We can put that muscle cell into a fascicle. We can put that fascicle into the muscle, and we can see how our body moves through space. So now that we have all the bits and pieces, what we can do next is we can now understand and work together to build back up so that we know how we move our body through space. All righty. That is it for the anatomy that we needed to go over. However, we're not meeting again until Tuesday next week when I'll get a chance to talk, and I want you looking at this material. So the last thing I want to do is give you an overview of what it is that we're going to be doing for the next couple weeks. So any questions on this anatomy before we move forward?
No.
Excellent. I love the confidence. Either people are asleep or they're confident in this material. Either way, I'm satisfied. Excellent. All right. So as I mentioned at the very beginning, this process of contracting a muscle is our longest physiological process. If you think about it, my goal is to move my right hand through space. That process starts, as we talked about, over in the left side of my head, on the left side of my brain, where I make that decision, I send that signal down to my hand, and eventually my hand contracts. It is one continuous process, right? And that's important enough to write down. This is one continuous, oops, forgot the T. From start to finish, I make the decision, my hand moves in place. However, it is the longest physiological process we're going to do. So to make it more easily digestible, we are going to break it up into 3 more easily digestible pieces. All right? And we're not doing this breaking up artificially or randomly. We're not just randomly deciding when we're going to break this up. We're breaking it up in a meaningful fashion. However, it's important to know that it's a continuous process. So when we talk about part 1, the very last step of part 1, guess what else it is? This whole thing's a continuous process. The very last part of step 1 is going to be what? Yeah, the very first part of step 2. Absolutely. So when we end part 1, the very last thing we do in part 1 is going to be the very first thing we do in part 2, because it is a continuous process. Excellent. So let's identify the pieces. And when we identify the pieces, we are going to name them. And I'm going to go ahead and write this out. I'm going to write this out here because I want to write it out a little bit different the way I would do it on the board. And then we'll do it with all the pretty pictures as well. Part 1 is called communication at the neuromuscular junction. The neuromuscular junction, as we see here in this pretty illustration up here, is where our nervous system talks to our muscle system. There we go, perfect. Now, for every one of our parts, we are going to have a goal. And the goal of part 1 is to irritate our muscle cell. Remember, as we talked about, our muscle cells are irritated. The way we're going to do that is we are going to make a conscious decision. And when we make a conscious decision, our nervous system produces a neural action potential. So our goal of step 1 is going to be to convert the neural action potential into a muscle action potential. So that is our goal of step 1. Step 1 is called communication at the neuromuscular junction. And the goal is going to be to convert our neural action potential into a muscle action potential. And once we've written that out all pretty on the exam, we can abbreviate that to, we want to convert a neural action potential into a muscle action potential, a NAP into a MAP. Part 2 is what we call excitation, contraction, coupling. Oops. Excitation, whoops. There you go. Contraction, coupling. Coupling is, of course, to bring two things together. And in this case, we just had our excitation. We have our muscle action potential, but we want to be contracting. We want to generate our force. And so that is going to be step 2. It's called excitation, contraction, coupling. Again, we need to focus on the goal of this step. And as we talked about, why aren't our muscles in a constant state of contraction? What stops our muscles from contracting all the time, being constantly in a state of contraction? Why isn't your myosin constantly grabbing and pulling on your actin? Right? Not just troponin, but absolutely troponin and tropomyosin, your regulatory proteins. So our goal of step 2 is going to be to move the regulatory proteins. And remind me again, what are we going to use to move the regulatory proteins? Moving those regulatory proteins is a wonky thing. So what might we want to use to do that wonky thing?
Calcium.
There you go. We're going to use calcium to move the regulatory proteins. That is going to be part 2. Part 2 is excitation, contraction, coupling, where we're going to release that calcium and use that calcium to move the regulatory proteins. And once we use that regulatory proteins, then part 3 will begin, and that is what we call the contractile cycle. Oops. Cycle. Of course, one of the key terms in that is cycle. What is special about a cycle? Does a cycle just go around once and stop?
No, it's continuous.
Yeah, it's a continuous process. Our contractile cycle is a continuous process. And this continuous process, the contractile cycle, our goal, if that's what we want to think of it in terms of, is to basically change the shape of the muscle. And really change the shape of the sarcomeres, I guess would be a better starting point. Right? And that is going to generate our force, or again, we can think of it, it's going to produce our tension. Oops. Now, to do this, we are going to move the myosin and actin. So part 3 is called the contractile cycle, but sometimes it is also referred to as the sliding filament theory. And both are acceptable terms for that, because basically what it is it's the myosin and actin sliding against each other, which changes the shape of the sarcomere and generates our force. It's what produces our tension. All right, so those are the 3 steps. I've written it here. I'm going to cheat and grab that and move that off to the side so it's out of our way. I can always bring that back if we need to, but that way we can do the same thing here as well. So again, step 1 is our communication at the neuromuscular junction. Again, it's our skeletal muscle. We are voluntarily controlling it, so we make a decision, and then our goal is to convert that neural action potential into the muscle action potential. Right. Once we produce that muscle action potential, remember our sarcolemma is specialized to produce and spread that action potential, and so that's what it's going to do. That is going to lead to our excitation contraction coupling, where our goal is to release the calcium from our sarcoplasmic reticulum and use it to move our regulatory proteins. And once we remove the regulatory proteins, now our myosin and actin can interact with each other. Lastly, we have our contractile cycle, or like we said, the sliding filament theory. This is where our myosin pulls on the actin, changing the shape of our sarcomere, which is what produces our tension. It's what generates our force and causes the muscle to contract. And so those are the 3 main steps we need to talk about. Now, if there was a 4th step, what would that 4th step be?
The muscle relaxes.
Right. Once we get that muscle contracting, we don't want it to contract for forever. So if there was a 4th step, we would say that 4th step is the relaxation of the muscle. And so here's what we're going to do. When we talk about these processes, these steps, we're going to do it both ways. We can talk about the relaxation muscle as a 4th step, where we can talk about how it relaxes all by itself. Or the other thing that we can do is that we can also, if we think about it, initiate these processes. So when we talk about turning these processes on, we can start the process, or what we say initiate the process. And for each of these processes, then we can also talk about how we would stop it or how we would terminate. So when we talk about each individual step, when we talk about communication at the neuromuscular junction, we will talk about initiating communication at the neuromuscular junction. We will talk about terminating communication at the neuromuscular junction. And to show we truly understand a process, we want to be able to modify it. So we'll also talk about how we can modify. So remember how I said of these 3 processes, 2 of them I guarantee are essay questions? Well, if you really think about it, we don't have 3 processes here. We have 9. Initiate communication at the neuromuscular junction, terminate communication at the neuromuscular junction, modify communication at the neuromuscular junction. Initiate excitation contraction coupling, terminate excitation contraction coupling, modify excitation contraction coupling. Initiate contractile cycle, terminate contractile cycle, modify contractile cycle. Right? 9 possible essay questions, And I guarantee at least 2 of them, possibly a third, will be essay questions on the exam. Sound like fun? You can see by all the thumbs up that I'm
No.
putting on the screen. No, it's not. Yes, it doesn't sound like fun, but the good news is that it won't be that bad. So it's not going to be that bad. And we will, again, it's not all, we're not doing all of that in the next day. We're going to be spending the next 3 weeks talking about all these things and making sense of it. So we will take our time, we'll go through it, and we'll make some sense of it. All right, so that is our game plan starting next week. And again, that gives you the weekend to not just look at this anatomy and make sure it makes sense, but to start reading these processes and seeing if it can make sense that way. That is everything I wanted to do from a lecture standpoint for today. However, as I mentioned at the beginning, there are some other things that I want you guys to be able to be aware of and you taking advantage of. Hold on one second, let me do that. Oops, and then I need to cancel that. So, okay, so wait, let's go back real quick. Any questions on any of this stuff that we just talked about, the game plan of what we're going to be doing? Again, I don't expect you to know it yet. That was just the introduction to it. We're going to go through it in detail, make some sense of it, and hopefully that will be good. The other thing that I want you to, so where was it? Here we go. This is what I want. Excellent. In our Canvas, under the modules, one of the things that I just added was a new module. In that module is where I'm going to put tools and resources that will help you with your studying for this. One of the things that is there already is this great website. Can you guys see this website now?
Yes.
Okay. This is a great website that is put up by Cosumnes River College. Again, their materials, their resources, not surprisingly, are very similar to ours. So this is a great resource. And notice what they've done, for instance, as we were to be interested, as we were talking today about the histology, we can click on the histology, and there we see some nice skeletal muscle tissue, some nice histology slides that they've provided for us. But notice as you click through it, they have the slides so that you can test yourself. And then they also have some labels so that you can turn on and off, so as you click through them. Now, are they necessarily going to have everything that you're going to be responsible for on here? No, not necessarily, but there we see our smooth muscle. We see some of those characteristics. So we have all of those things. There's our cardiac muscle, and it can show some of the histology. There is a fair amount of histology you're going to be responsible for on this. But the other thing that is going to be a great tool that's going to help you, and eventually get out of this, perfect. Let me actually make this add that to my desktop. Excellent. All right. What the other thing that you can do, for instance, if we look at the limbs, we have pictures of the models, and they have the exact same models that we have. So as we look at these things, you can challenge yourself on that. Notice when you click the button, it shows you the labels. So you have this resource, which is a great resource to be able to look and study the models. So you can see all these things. You can challenge yourself with them, and then you can click it to see the tags. They have lots of different examples, lots of different ones. Now, again, is every single muscle on here something that you're going to be responsible for?
No.
No, absolutely not. However, you do have a list. And on that list, you can see. So for instance, when you look, you'll see that the extensor pollicis brevis is not one of the muscles that you're going to be responsible for. So feel free to ignore that one, right? You don't have to be worried about and responsible for looking at that one. That one's going to be okay. You don't have to necessarily know that one. But there's plenty of really good things on here as well. Remember the other resource, and so hold on, let me come back here, do that. The other resource, and it may take me a second to get there, is in your Master in A&P. So let me get to the Master in A&P. So there isn't any reason why you shouldn't be able to study these things at home. This is in some ways actually going to be easier than it was for the bones to be able to do this. All right, let's see if this works now. All right, do you see the study area here?
Yes.
Okay, when you guys go to the additional study, under lab tools, and then again, there's plenty of other things. There's art labeling, there's practice tests, there's all great things here. But in this case, what you're going to want to do is your practice anatomy lab. Let me know if you guys aren't able to see these things as they pop up. You guys see that?
Yes.
Okay. They have the humor cadaver stuff, which is fun and interesting, but what's going to be more meaningful for us is the anatomical models. Notice if again we go to the muscular system and we go to the upper limb, just like on the CRC site, we have pictures here of the models. Notice I can move my mouse over them and highlight individual muscles, or there's a button up here where I can show them that way as well, and different views, so I can go through them and see the things there or hide the labels and test myself. There is a quiz, so when I'm ready, I can identify a muscle and try to identify what it is, and I can guess what it's going to be. And again, it's not always going to necessarily be something that is on your list, but it's still something that you sometimes could be responsible for, so make sure you're doing that, and if it's not one you're responsible for, ignore it. There is a lab practical that you can take where you actually have to write in the answer of what that is, so it can be challenging and it can be a fun way to study this material that way. However, what I love about this PAL process, and it's a CD that can be purchased separately if you don't have it as well, is notice up here at the top, there's where it says animations. Can you guys see that?
Yes.
Excellent. When you click on that, notice again, if we go to the upper limb and I go to the bicep brachia.
Biceps brachii is the large two-headed flexor muscle.
Can you guys hear that?
Yes.
Okay, perfect. I'll let it play then.
Located in the anterior compartment of the arm. The origin of the long head is from the supraglenoid tubercle of the scapula. The origin of the short head is from the tip of the coracoid process of the scapula. The two heads insert by a common tendon onto the radial tuberosity and by the bicipital aponeurosis, which blends with the deep fascia on the medial aspect of the forearm. Biceps brachii flexes the forearm at the elbow supinates the forearm, and weakly flexes the shoulder. It is innervated by the musculocutaneous nerve.
So notice it has the attachment points, the origins and insertions you're responsible for, the actions. It isolates the muscle and shows it to you. It is a great resource to help you to study this material. So I strongly encourage you to take advantage of these resources that are available to you. Hopefully you purchased the Modified Master in A&P. You can see it that way. Hopefully you can, again, use that CRC site because that's a great resource. So there are ways, even though you don't necessarily have the time in the classroom. Which also reminds me, Jeff, I just sent an email out where Jeff posted that he is going to be doing virtual Friday labs So he's going to be available on Friday for a virtual lab on Zoom. He's got some limited models with him, but he can answer questions and help you guys with this material as well. So you are absolutely positively responsible for this material. You guys will have lots of resources that can help you to be successful this way. All righty. Questions on any of that?
I have a question about the recording. Is this going to be uploaded on YouTube or onto Canvas?
YouTube. So
Okay.
in talking with some of the other instructors, so I was trying to get it to do it because there is a way to get it to save it to Canvas and do it through Canvas, so that where we have these, you have access to the meetings and the videos there. But what I found is that there were restrictions on the size, there's restrictions on the timing of it. And the other problem is because most of the Zoom stuff is cloud-based, even through the Confer Zoom. A lot of instructors are having problems because of just the massive use that Zoom is having issues with right now. So it's taking some people days to spool and get up there, and still having problems with it. I have had a much more successful time getting these videos up onto YouTube, and so the YouTube will be much more advantageous. I don't actually know the YouTube page, but if you just do a search for Daniel Slutsky, there can't be that many of us, and the same that synapse picture that you see that is my label on this is my label there as well. So if you just do a YouTube search for Daniel Slutsky, and if someone can get the link real quickly and post it in the chat, that would be excellent as well. But you can do just a quick YouTube search for me. It's also in the email that I sent. It's also in the announcement that I posted in Canvas. So you have all those ways, and that's the easiest way to get there. It takes about an hour for both Zoom to convert the video for me. Perfect, thank you. Convert the video for me, and then also then for me to upload it and get processed by YouTube. But as soon as we're done with the lecture, that's what I do. So usually about an hour, hour and a half after our lecture, it will be up there. Again, that's just based on the first 2 times I've done it. But it was successful yesterday. It was successful on the test one. So knock on wood, it should be successful for these as well. So I think that is a more easily accessible way of getting that material up there, and it seems to be working. So we will do that.
Okay, thank you.
No, thank you. Any other questions? I love these questions. Great questions. Again, this is a new process for all of us. As I've been saying from the get-go, I know I did not sign up to teach an online class. I know none of you signed up to take anatomy and physiology online. So this is a very odd and unique situation we have found ourselves in. And it's going to be a learning process, and there's going to be growing pains for all of us. I was incredibly impressed with the success that you guys had on the exam. I haven't graded the exams yet, so I don't know how that went. But for the fast turnaround that we had to get that set up and going, at least from a process standpoint, there were only a few minor bumps and bruises along the way. But for the most part, the process was successful. And then if we find out we're going to have to continue that, then we can refine that process more, so it'll be hopefully even easier to work with in the future, and yet still maintain the integrity of the class and everything else. But for the fast turnaround, I was incredibly impressed with how great of a job you guys did with that, with only just a few minor problems. So I was very, very happy with that. And so hopefully, like I said, knock on wood, hopefully these lectures will continue to work, and this will be something that will be successful for helping you guys to get this information.
Would extra credit be available for the open lab still?
My concern with that from Jeff's standpoint is I don't know how he would be able to keep track of who was there and how long they've been there. So I don't know if that's something that can practically be done that way. Knowing that that is the case, though, I think I will try to be, maybe we'll try to find some other ways to add. There are a lot of activities in your mastering A&P, whether it's, the fill in the blanks or the labeling activities, or there's crossword puzzles or things like that. Maybe there's some way of that, because again, really, the 15 points that a student would get for going to open lab for 15 hours usually doesn't make or break somebody's grade. Most people don't fall 13 points shy of a B or 13 points shy of a B, and, I mean of an A, and if only they'd gone to open lab, they would have gotten those 15 points they'd put over the top. Every once in a while, does someone fall a couple points short? Yes, but it's not very often. So the points aren't as major of a deal as it is for the 15 hours that you're going to spend studying the material. If you spend 15 hours studying the material, you're going to know that material much better than you would have otherwise.
Right.
There's a mandatory quarantine that takes effect tonight. Would open lab still be open? Yeah, but no, remember the open lab is a virtual open lab. If you look on the email that I sent, he's going to do it via Zoom. So it's going to be Zoom open lab. So it's not actually on campus. So please do not go onto campus. It is just a virtual online open lab that he is doing.
Have you heard anything referencing whether or not they're going to start finding people who are violating the quarantine? Like, you're not supposed to leave your house for like any reason other than something that's mandatory, like going and getting groceries.
I know they're doing that in San Francisco, for instance, which is a much more highly impacted area. I don't know if Placer County I haven't heard anything about that in Placer County or Sacramento County, these areas. I think the fact that we're spread out a little bit more may mean it's a little less likely, but again, it seems like information is changing daily. And I think one of the ways that we personally see that is that the Los Rios School District is incredibly non-committal about when this is going to end or how long this is going to go on or anything along those lines. They refuse to provide any information. They shut things down on campus almost immediately, right? Whereas many campuses went more hybrid or allowed labs to come, or they took more time to transition. They shut us down immediately, and they refused to give us even a tentative date of when it comes back. So I honestly don't know, because there's going to be obviously the decisions that the county makes, but then ultimately it's going to be the district that decides what we're going to be able to do. Now, of course, if the county says, don't leave your houses, then obviously they're not going to be able to start school back up. But I don't actually know. I know exactly as much as you guys do.
I did hear that they were saying, I know for the kids' school district, they're saying for the rest of the year, kids are out. So I'm pretty sure that they're going to probably do the same thing for
Well, see, but again, that is very school district related, because my kids, one of my kids, I'm in Roseville, and the Roseville Joint Unified School District, where my high school kid goes, still says that they're going back April 15th, basically, or whenever spring break is over. Now, they admit that these things are fluid, and just like when they close the school, they happen very rapidly. There can be changes as well. But based on the information they're getting from the county, they say that they are going to be starting back school up again after spring break. Now, again, the fact that they're saying that is completely meaningless, because things change literally on an hourly basis. But other schools are saying, yeah, we're just closed for the year. They're saying, no, we're going to try to come back if we can. So again, everybody's different, and that's part of the problem. If we had strong central leadership, then maybe there would be some federal rules would be set and everybody would be doing the same thing, but that doesn't seem to be the case. So, the right hand doesn't know what the left hand's doing, and everybody's kind of making it up as they go along. But for now, this is our plan. What we have here will work through spring break. We don't have another exam till after spring break. Our exam right now is scheduled for the Thursday after we would come back from spring break. So we can certainly continue to do what we're doing from now till spring break. And when we get closer to spring break, if it looks like it's going to extend beyond, then we have spring break to do things like, make sure everybody's got webcams that work, make sure that the tests are made properly, and all those types of things that would be necessary for that. So we're not going to be as rushed to have to try to do these things in 2 days the way we had to do when we started this. So the good news is that if this goes longer, and I'll keep my fingers crossed that it won't, but if this goes longer, then we'll see what goes from there. But at least for the next couple of weeks, I think this is going to be our game plan. Great question, you guys. Any others? All right, well, that's all I've got. What I will do is I'll leave the recording going for 5 more minutes, just in case anybody else has any other additional questions or anything that they want to ask. So I'll stay here for 5 more minutes, but then at 3:20, if there haven't been any further questions or if there isn't any more information, then I will end it at that point. Because like I said, that is the material that I wanted to go over for today. And then make sure you guys do your best to prepare. One of the things that I did do, last thing, as I mentioned, starting next week, we are going to start doing our lab stuff as well. Our lab stuff is learning the muscles, their actions, their origins, and their insertions. You are responsible for knowing your bone features from the last section and using those. So one of the things that I have on Canvas, and it was locked till today, but I unlocked it today, so you have some time to play with it, is some illustrations of the skeletal system, where we'll be able to draw the origins and the insertions. And one of the things that I'll tell you as you look at the material in the textbook and everything else, by convention, which of course means just the rule that somebody set up and they all decided on, when we're drawing the origins and insertions, origins are going to be in red and insertions are going to be in blue. So, so if you want to start looking at those and thinking about those, we'll start drawing those and talking about those next week. All right, so like I said, I'm here for any more questions till 3:20. Otherwise, thank you guys very much. Hopefully this was at least hopefully partially informative, helpful, something like that to help us to get through this. Hopefully this worked well. All right, thanks guys, and have a good weekend. Study hard, and I will see you next week. Like I said, I'll stay here for a few more minutes if anybody has any last minute questions. Otherwise, have a good weekend. Stay safe, stay healthy. All right. That's it.